Okay, so now we are live and would like to welcome all the participants who have just joined in for the webinar 10 regulated bioanalysis session for the monthly theme regulatory challenges, learnings and opportunities. And another 5 minutes, not five, I think 4 minutes, and we'll get started. So just hang on there, keep your lines muted, and we'll start at 8:45. Thank you. All right, so let me just welcome all the participants, and I think still they're joining in, but since it's already time, so we'll get started, and maybe they will join uh, while the introduction is going on. So I think let me quickly. Okay. So Patanjali, if you can just confirm or any of the organizers if my screen is visible.
Yes, it's visible.
Yes,
Can you hear me?
Okay. All right, great. Thank you. So once again, a warm welcome to all the participants. APA India 2021 webinar number 10 in regulated bioanalysis session. And for the month of September, the theme is regulatory challenges, learnings, and opportunities. So as always, we would like to thank our organizers, as we have discussed earlier also, that we have organizing committees for each of the sessions. And each committee is headed by presiding chair and the chair-elect. So the chair-elect will resume their office for the next year, and presiding chair is for the current year. So we would like to thank all our organizers, especially presiding chair and the chair-elect for different committees. We would also like to thank the organizers from each committee, like for regulated bioanalysis, discovery bioanalysis, biotransformation, and chemistry, manufacturing, and control. So basically, all these organizers, we meet once in a month, and then we really discuss about the agenda and the speakers selection, topic selection. So the entire credit goes to this esteemed organizing committee members of each committee for having such a wonderful agenda on board for APA India 2021. So a big thank you to all our organizers. Without support from sponsors, not possible to do an international conference consistently for the last 10 years or so. So a special thanks to our sponsors, and would like to mention that most of them are with APA India chapter for almost a decade now. So a big thank you to our sponsors, Shimadzu, SciEx, Zepsci, Phenomenix, Thermo Fisher, Agilent, Syngene, USP, Netco Pharma, Vida Clinical Research, Bristol Squibb, Zoetis, IVL, Invitroid Met Labs, Origin, Nanotemper, and Loris Infosystems. So thank you to all of our sponsors. This year we have collaborated with the Life Sciences Sector Skill Development Council. So we, just for people to know, LSSDC is a national level certification body under Ministry of Skill Development and Entrepreneurship. So thank you to our collaborator. A very quick couple of announcements, which we do always, like just for the reminder. We do Kindle draw at the end of the webinar. So would request all the participants to be there throughout the webinar, and to participate in that Kindle, you will have to participate in the webinar poll, which we do after all the 3 talks in a webinar are over. So would request attendees to please participate in that. Up till now, we have done 9 webinars starting from June till date. And this is the list of all the winners of the lucky draw, Kindle draw. So I would like to congratulate all of them. If you have any question, if you have any questions or queries about any of the talk which is going on, feel free to post your queries on the Q&A tab at the bottom of your screen, and moderators will pick up those questions and they will pose the same questions to the speaker after the talk is over. Yeah, and during the webinar, if your queries are not clarified or are not addressed, we request you to write to bostondb21@gmail.com, so that we can reach out to the concerned speaker and get back to you with your answer. As I mentioned, do participate in the poll at the end of the webinar. And also, webinar talks, so right now they're live streamed on YouTube, and they will also be available on a YouTube channel of Boston International. And then you can go back and review the talks also. So with that, I think without further ado, let me get started. So once again, today is 11th September, webinar number 10, regulated bioanalysis. And let me straight away invite the chair, Patanjali, for his inaugural address. Over to you, Patanjali.
Hi, thank you, Deepak, for the kind invitation for the APA India September session, the monthly theme being regulatory challenges, learnings, and opportunities. Good morning, good afternoon, good evening, wherever you are. I'm Patanjali Kariyala. I've been with BMS since last 29 years, currently an Associate Director for Outsourcing Operations and Management Group. Today, the topic relevantly says regulatory challenges and learnings and opportunities. Over the years, the regulatory landscape has changed throughout the world, especially FDA, EMA, then NMPA regarding China, Korea Health Authority, then Turkey Health Authority, China Health Authority, then we have Health Canada Authority. All these agencies have come up with the recent regulatory changes, and to keep up with them is a challenge. And most of the bioanalytical labs, which are managing the work, keep up the regulatory updates through their changes in the SOPs to meet the requirements. And as the final product, every study is the CSR, which is clinical study report. For me, that CSR translates into compliance, science, and regulation. So that CSR has to be a product which meets all these requirements, so that the study or the data which is coming out of the study truly represent for the patient-centric dosing or the regimens required for furthering the compound in the clinical studies. With that introduction and with that intro, I like to invite Clarinda Islam, who is part of Samru Bioscience Canada, who will provide us the overview of the global regulatory landscape, advancing biosimilars and development. If we can project that, yeah. Clarinda Islam is the Vice President of Project Management and Regulatory Affairs at Samru Bioscience Incorporation. In her current role, she is responsible for managing both the production and R&D operations. She is also responsible for the management of large-scale client collaborations and projects. She oversees the team's close collaboration with clients to develop bioanalytical strategies that support pharmacokinetic and pharmacodynamic immunogenicity, biomarker, and biosimilar studies. Previously, Ms. Islam has worked in CROs and clinical laboratories. She has over 15 years of experience in bioanalysis. She brings a comprehensive knowledge of regulated bioanalysis and hands-on operational experience in delivering scientific and service excellence to pharmaceutical and biotechnology clients through her experience in bioanalytical science at biotechnology companies and CROs. Without further ado, I welcome Clarinda Islam for the first topic. Thank you, Deepak, and welcome, Clarinda.
Thank you.
Sorry, Deepak, go ahead.
Yeah, please, over to you, Clarinda. The floor is all yours. So please share your screen and get started. Thank you.
Great. Thank you, Patanjali, for that wonderful introduction. Let me see if I can get this back. Okay, already I am running into technical difficulties. Deepak, how do I maximize my screen? It seems to have minimized it.
Actually, on the right-hand side, you will see just the minimize option. That's. Did you share the screen first? I mean, did you share the screen?
No, that's what I'm attempting to
okay,
do.
Okay, no worries. So just double-click anywhere on the screen or on the top. You can just move your cursor. You will be able to see all those options to share the screen. Yeah.
Here we go. Thank you. All right.
Okay.
I'm ready to do that. And share and.
Yes, we can see, and just you have to do the slideshow. Yeah, you have to do the slideshow. Yeah, perfect.
Is it visible?
Perfect. You're ready to go.
Excellent. Okay. Well, thank you for that. Sorry about the little hiccup. Thank you again, Patanjali, for that introduction, and thank you to the organizers for inviting me here to share a little bit about my experiences and Samru's experiences in general in the arena of biosimilar development. and supporting our clients and partners in advancing their biosimilar products into the development and in eventual approval. So just as a little overview, I'm going to be touching a little bit on some of the key things that have developed over the past several years as these guidances have solidified and have been finalized, and industry as a whole has had a better grasp and understanding of the implementation of such things. We're going to touch a little bit on the pharmacokinetic assay and the things that are involved in the regulatory landscape around the guidances on that, as well as immunogenicity, the biomarker PD testing, and last but not least, in the software systems, as more and more our data is being assessed, compiled, and delivered in an electronic format. So there are definitely some regulatory requirements around that. So just kind of going into it a little bit, our biosimilar testing, the regulatory guidance. So the EU was the first to establish that pathway, giving us a pathway for having these biologics or the similar biologics, giving a pathway for approval via the EMA guidances. Since then, since 2005, many countries have since followed suit. And there is, at the heart of all of these guidances, we can kind of distill it to the essence of it, being the demonstration of that similarity between the reference product and the biosimilar. And what does that really mean, similar? It can be interpreted in a lot of technical details, but it really comes down to, are they performing in the same manner, and is it as safe or safer than the reference product? And how do we assess that? It is, to borrow a phrase from the FDA, the totality of evidence. A lot of regulators look at the whole picture of this, but the primary key data that where all of this evidence comes from is at the heart of it is the PK, the ADA, and the PD biomarker data that is generated. So let's start a little bit into the pharmacokinetic assay. So at the heart of the biosimilar analysis and biosimilar comparison, we're comparing a reference product to our intended biosimilar, to our new product. And one of the first things that you do is do a standard curve comparison. And it is important to utilize statistical approaches so that you can ensure that this comparability is the same. And then you do your second tier comparison using your validation samples or your QCs. However, one thing to note about that, even when the QCs meet the standard criteria, and that's typically our plus minus 20% bias, our CV at 20% or less, if there is a trend observed, the regulators will likely ask for an explanation. So it is just as important to know very early on and to monitor your QCs to see if a trend is being developed. A little bit more on the comparability of reference product and biosimilar is minimizing the variability in the preparation. And that applies to both the standard curve and the QC. We want to ensure that the variability that we're seeing, if any, would be due to the differences between the reference product and the biosimilar, and not due to preparation or analyst variation or any of those other inherent variability that we'll see in any of the typically in ligand binding assays or other type of technology that we're using. But technology could also be the solution to this, minimizing the variability. Utilizing automation, for example, for preparing standard curves or QCs. And it's important to use the same script. So utilizing the automation script, as shorthand for using the same type of programming or same type of preparation scheme whenever possible. Automation can go very, very far in minimizing that variability across your different preparations in using a PK assay. Another key thing to kind of think about when you're establishing your PK assay for biosimilar testing, the use of a surrogate matrix. There are times when a surrogate matrix has to be considered if the drug of interest, it has an endogenous counterpart. But we do have to justify the use of it. and the assay should have a sufficient MRD to demonstrate that that surrogate matrix can quantitate the samples in the study matrix accurately. So there's several different ways of assessing that kind of accuracy. There's your spike in recovery, dilutional linearity, or using or testing a parallel via parallelism. Now let's touch up a little bit upon the reference standards that are being used in the pharmacokinetic assay. So we need to always have a very good idea of characterizing the reagents that we use. And one of the heart of one of the reagents that's the most critical in our PK assay is our reference standards. So we need to have a good understanding of, there are times where you will have, instead of an expiry date, you will have a retest date. If you are early on enough in the process, there is a retest date, and it's or sometimes it is unclear what the reference standard might be in the early part of the process. So a certificate of analysis is always required, typically required. If one is not available, however, an equivalent alternative to that should be acquired and on record, so that we are ensuring that from batch to batch or lot to lot or production lot to production lot, that we are ensuring that it's the same product in the terms of quality, in terms of purity, and in terms of performance. Another key thing to kind of keep in mind with our pharmacokinetic assay is the sensitivity. So the sensitivity has gotten more and more important over the past several years. As our technology has improved, we've been able to really push the boundaries of the sensitivity of our assays. However, we also have to have a very good understanding of when it is necessary or critical to do that. In some cases, during PK comparison, a sponsor may use a lower dose than the therapeutic dose. And also the suitability of that sensitivity must be well understood. So for example, teriparatide, it requires anywhere between 10 to 20 picogram of sensitivity. There are now instances where even beyond 10 is being evaluated and considered. So it has to be well understood for the purposes of what your PK assay is going to be doing in terms of its fitness for the study. The next thing to consider is our dilutional linearity. So for a lot of our studies or a lot of our assays, it does require a significant dilution of samples. So it's something I think everybody is fairly aware. It should be tested at the appropriate level during the validation. Just a reminder that samples should not be diluted any beyond the dilutional linearity that has been established during the validation. And the latest guidance from the FDA does require the use of dilutional QCs if dilutional linearity was not demonstrated during the validation. So if there are some rare instances where the range is adequate, your calibration range is adequate enough to cover what is necessary during for any particular study. However, if there happens to be an instance where a dilution is required for samples, you do have to add that dilutional QC into your assay if it was not demonstrated during the validation. So this is a quick snapshot of our requirements and the industry practices that have been kind of in place, right? And as these guidances or requirements have been formalized over the past several years, and some of it deriving from already established industry practices, some of it deriving from just a more understanding of what is required to demonstrate biosimilarity for PK and biosimilar requirements for ADA, and in this case, we're particularly concentrating on PK requirements. So this is just a quick snapshot of how what those variations are, perhaps, that there are. So in a lot of instances, they do overlap quite a bit. And if you are always, if there is ever a question, I would always recommend, well, then go for the more stringent requirement. So that way, there isn't any catch work that's going to be required later on. Just to touch on a little bit, I know actually most of my focus and most of my work for those of you who I've worked with before regards more or revolves more around ligand binding assays. However, there are now getting more and more instances where we are exploring the use or actually using LC-MS/MS methods for doing drug measurement or PK assays. So it's perfectly reasonable to use this methodology for doing PK assays. A justification for the method choice will need to be included in the report. And just a couple of things to remember, which is typically something that we would consider for any PK assay anyway. Endogenous counterpart interferences should be evaluated if it's present. So for example, showing that PTH1 to 84 does not cross-react with PTH1 to 34. It is also important to illustrate that the validation samples that you use during validation have been treated the exact same way that you intend to use your test samples or your study samples. And then it's just ensuring that the validation report is submitted with the study report so that the methodology used is well demonstrated and well described. Now, I'm going to touch on a little bit on commercial or diagnostic kits, but I will have a little bit more to say on this a little later. But there are a lot of instances where there are commercially available kits for the use of for use in PK assays. Just a couple of validation considerations for using these. Again, you need to evaluate the suitability of the reference material. And then also assess your calibration range or the number of calibration standards. It may have to be expanded. There are times where some of the calibration points, they run a little short, and it has to be evaluated just as if we were using any fit for purpose method that's being developed and validated. The QCs that are used, they have to be prepared independent of any kit supplied QCs. There are times where some of these kits come with ready-made or already prepared QCs, but it is incumbent on us to independently prepare QCs that reflect the matrix that is being intended for our study samples. If there is a requirement or a need for a different matrix, it does need to be justified and demonstrated during validation. And last but not least, a lot-to-lot variability. A plan for assessing this variability should be in place and should be evaluated, especially for studies that can run, fairly long, and that would probably, last beyond 12, 18 months, which is sometimes well beyond the established, expiry dates of some of these commercial kits. So to link all of this to a little bit more about our biosimilar testing. So this kind of progression should be familiar to a lot of people here. It does follow our typical development phase, the validation phase, and in-study validation or sample analysis. However, just a couple of different things that we would be doing here when we're doing biosimilar analysis. Because of the nature of this, we have to confirm the comparability of the reference product and the biosimilar. So we typically would do that during the development phase to do the curve-to-curve comparison. And then once it's established or once it's been demonstrated that both of these products perform similarly, then we can move on to the validation phase only using one of them, and usually it's the biosimilar product that's used. But then during validation, we demonstrate that further by performing accuracy and precision using both the reference product and the biosimilar product. So here's just a small illustration or a recommendation that can kind of give you an indication of how we might perform them. Again, the calibration curve comparison is typically done during method optimization or method development. And its purpose of it is, again, to evaluate the similarity of the biosimilar and innovator or reference product curves. And it is recommended that we do this to 2 to 3 runs. And for each calibrator concentration, then we can just take an average response across the 3 preps. So here is a representation of how we might do this comparison. So you can see here We are using both, we are plotting both curves on one graph to kind of, and do the appropriate calculation to do the calibrator curve fit, and then do some additional statistical calculations to compare the two curves. So one of the things that we use here, as you can see, is the F-test to evaluate the parallelism, or how closely similar these curves are. If they are found to be parallel, then we move on to using the EC50 ratio. If it shows that the curve is not parallel, obviously one of the first things that you do, investigating the root cause, and repeating if necessary, if nothing else, just to confirm that the curves are not parallel. There may be a time or instance where when you go into validation, you may have to use two PK assays to measure both the biosimilar and the innovator independently once you go into sample analysis. One other thing to note during this calibration curve comparison, once the EC50 ratios are calculated, another measure of showing this comparability is that the EC50 should fall between 0.8 to 1.25. Now, again, this will have to be evaluated specifically for the assay that you're using, as criteria may not be always appropriate. It could be dependent on how the curves are due to the differences in the asymptotes, or where your upper and lower limits would lie. But in general, ensuring that the slope and the asymptotes of the biosimilar and the innovator curves are similar gives you the necessary assurance and a demonstration that you can move on to your method validation with the one assay, and that you can be successful in the method validation. So once in method validation, then we would be comparing your validation samples using both the reference product and the biosimilar. So here is a recommendation or an example of a balanced plate design. As you can see here, once we've demonstrated the comparability of the biosimilar and the reference product for the calibration curve, we can proceed with just using the biosimilar as our calibration curve, and then have both types of validation samples read against that curve. And you want to kind of balance it out across the plate. And again, using our typical to do the comparison for the quality controls, the assay parameters we would typically use are our recovery error, both inter and intra, and our percent CV, and then ultimately our total error, with special consideration for the ULOQ and LLOQ concentrations of our validation samples. So that just kind of wraps up what I wanted to touch on pharmacokinetic assay. So for a lot of it, it is straightforward and fairly straightforward, and there's nothing entirely new from our experience in doing PK assays from before, with the added additional responsibility on us to demonstrate that biosimilarity of the curves as we do the method development and optimization, and then against demonstrating it during validation by using both products for establishing accuracy and precision. Now, going on to the immunogenicity assays. Immunogenicity can be, again, quite complex, with especially as in the past several years, we've through a combination of just experience with the regulators, our increasing development and improvement in the technology, the reagents, detection methods that we've been using. So we are able to really characterize our immunogenicity assay. But I kind of want to highlight three major things that really cut to the heart of what makes a very successful immunogenicity assay that we use for our biosimilar development. So first thing that I kind of want to highlight, it's all about the positive control. We always recommend we generate a specific positive control to the biosimilar. There is justification to be made for using commercial controls or controls that have been developed against the innovator. However, in order to really demonstrate really demonstrate the city part of your a possible biosimilar product, it is always recommended to generate a specific PC to this. We characterize it in the typical normal fashion that we do any critical reagent. Generating specific positive control requires us to have a good awareness of its specificity, of its sensitivity, and so on and so forth. Couple of things on a couple of the parameters we usually establish or evaluate during validation. Long-term stability. So the EMA doesn't quite require long-term stability of the positive control. It's typically, sometimes it's interpreted as the long-term stability of your samples. FDA, it's not so clear, but it's, again, it seems to be that the stability is around the suitability of your QC samples rather than the stability of the study samples, right? So one thing though that should be kept in mind on this then, and where it becomes important is the trending, specifically the trending of the LPC, where you are pushing the sensitivity or the boundaries of your assay, right? Demonstrating consistent performance may be sufficient, but it should also be something that you need to be aware of where your LPC is trending. And I'll kind of illustrate a little bit how this will come into play in importance once you get into sample analysis. Another thing to keep in mind is the variability of your LPC inhibition. So when you do the confirmatory portion of our tiered approach to ADA, it is expected to see a bit higher variability at the lower end of the inhibition curve. It could also be possible to that you may need to evaluate or actually use two separate LPCs. One that you use for screening and one for confirmatory. So this is something that you do need to keep an eye out on when you're doing your method development, and also when you go into method validation. It could be necessary that your LPC levels will be different between the confirmatory and the screening. The next thing that is kind of critical to immunogenicity assays when it comes to biosimilar testing, it's all about the comparability. So while all of the things that we typically assess during immunogenicity, so our cut points for screening, and cut points for confirmatory, the selectivity, the sensitivity, the drug tolerance, based on a priori criteria, you have to know what the assay is actually doing. It could be possible there will be significant scrutiny on the slightest differences of what you'll see during validation. And again, we can I'll touch a little bit on this where this becomes fairly critical. But here, while I will also emphasize the use of statistical approach for comparing curves, or in this case, our signals from our controls, as opposed to eyeballing it, right? So we can typically, when you're doing a quick review, you can have a general sense of what that is, but we would never rely on that. We would really use our statistical methodology in order to demonstrate this comparability. And how do we do this comparability? It's all about the statistics. We have to understand the statistical methodology that's being used to generate our confirmatory cut point, our screening cut point. How do we evaluate our outliers, whether it's the biological or the during an on-site audit, a regulator can definitely go all the way back to reviewing or asking about our method development data. And included in that would be our comparability data. It could also be possible that they might ask what the comparison is when you use a cut point using all the data, or after you remove the outliers. Could be a question around that. And again, I'll touch on the trending of the low QC, especially as the low QC, because this is where you would see the most variability if it would be apparent. And finally, the use of an in-study cut point. So while you're in sample analysis, it could be necessary to demonstrate, or they might ask that you establish an in-study cut point and compare it to what you had generated during validation. So having said that, the complexity of having to manage all of that during an immunogenicity validation, you can see this is just the typical things that you would evaluate during immunogenicity. It does take quite a bit of organization and computing power to do this. And, when we were all starting to say, I'm sure everybody has that experience of how disparate this could be sometimes. And one of the things that we've been able to utilize over the past several years is a statistical and a computational tool, AGRIS, in harnessing all of that and consolidating all of that into one key component where we can do a lot of these parameters and just consolidating all the data, and more specifically, most importantly, the all-important screening and confirmatory cut point calculations. So earlier you heard me say it is definitely important to use statistics, the power of statistics, in order to establish this. So the approach AGRIS uses is definitely from the FDA guidance, as well as some of the white papers that have been generated around that, with regards to how we evaluate the data for screening cut point and how to calculate and establish that. So this is just a general illustration of how screening cut point is generated, and separately how confirmatory cut point it is generated via the use of AGRIS. So you can see that where the statistical evaluation is being flow, I should say, the evaluation and flow is being deployed for both screening and confirmatory. And one thing I had already mentioned in general, it is generally highly recommended to establish an in-study cut point, especially when your study population for your particular, for any particular study is different than what was used during validation. So this comes into play when, once you're moving from your healthy volunteers to your, diseased volunteers. And it is becoming fairly routine to have regulators asking if you have established an in-study cut point. So it is important to demonstrate that the validation cut point is appropriate for the study sample analysis. And then you can kind of have that comparability and can justify it. If there is statistically no difference, great. If there is, then it becomes even more important to ensure that the appropriate study cut point is being used for whatever study is being performed. So a little bit on where all of these come together. So I'm going to kind of brief overview a little bit of a case study here. This was generated from an on-site audit of a study. It was a safety study comparing the reference product with a biosimilar. So these are from healthy volunteers for this safety study. But a couple of key things to note this. So the validation was performed over 4 weeks. However, the sample analysis is actually performed about 4 to 6 months after the validation. So from start to finish, about 4 to 6 months. So during method development and validation of that, so both the biosimilar and Neupogen, the sponsor, that is what is utilized, the direct a direct method, direct binding ELISA is used. The positive control, however, that was used in this particular study was a commercial antibody, meaning that it, so in all likelihood, this antibody was generated against GCSF. It was recommended that positive control specific to the biosimilar be generated, but again, that does take time. And due to the time constraints, the sponsor was not able to wait or didn't want to wait. So the anti-G-CSF, that commercial antibody, is used for both the initial validation and then later on during once a rabbit antibody was eventually generated, then a bridging was performed. However, the sponsor did start the study prior to the validation being completed, again, against the recommendation, because typically you want to have a well-characterized assay, and you would want to start this and really be completed with your validation about 3 to 6 months before your study start. I understand that's not always feasible, but it's always a goal to aim for. Now, earlier you heard me say that, so one of the things, it's all about positive control. So choosing the right, in this particular case, it's the right LPC. So the LPC was set at 100 nanogram per mil. And when it turns out is that when we did some statistical analysis on it, this was actually more of a high negative control. When the eventual rabbit polyclonal was used as the positive control, that concentration was deemed to be a high negative control. Having seen that, another positive low PC, ALPC, alternative low positive control, was used at a slightly higher concentration. However, we did keep the 100 nanogram per mil PC and still called it the LPC. So this was in essence a mistake. You can have multiple levels of controls there, but really you should be clear on what that low positive control should be. And then the other thing here, again, this goes into kind of the heart of showing that comparison, showing that equivalence. A separate validation report was done. So it looked like independently the biosimilar, it looked like the biosimilar was not really compared against the reference product, or in this case, the Neupogen, right? We did not show an assay comparison and equivalency right off the bat. And so this is kind of like just a snapshot of what that might look like. So these are the different, we're concentrating a little bit here on the cut point that was generated and the correction factor that was eventually used. So you can see on some of those red highlighted things where that percent difference is that is falling. And so this is kind of what where we want to really emphasize that comparability needs to really be established before you even start your sample analysis. And as well, establishing, characterizing the positive control within your method development and showing it or demonstrating it during your validation. So the actual results of the sample analysis. So using the validation cut point, the 22 samples were screened positive. Using the in-study cut point, however, 27 samples screened positive. Now, in this case, the particular types of samples, or those particular samples, were deemed that because of its eventual result, it would have been unlikely that these 5 additional positives would have been positive during confirmatory. So it is a little bit lucky in that context. However, this I include this to really emphasize the context of having to really understand where that cut point is. And again, as more and more auditors and regulators ask for it, is to the importance of setting an in-study cut point or a study-specific cut point. So to recap all of that, really our lessons learned there. It's all about the statistics. It's all about how definitely when you start establishing that cut point correctly, and how you do that is by using the most appropriate statistical approaches for the data set that you're generating. Choosing the right positive control. We always recommend now, that it be generated specific to the biosimilar. And after having generated that, to have it be well characterized in terms of its performance, its affinity, and then at the end of your development and optimization is to having or choosing an appropriate sensitivity for that positive control. And lastly, demonstrating comparability, recommending the validation start 6 to 9 months prior to study start so that you have a very good understanding of your immunogenicity assay. And actually, this really doesn't apply to just immunogenicity. It will also apply to your PK and PD or biomarker type assays. And lastly, using that in-study cut point as more and more regulators are kind of looking for you to have assessed that. A couple of other points of the ADA on ADA or immunogenicity. So the PK data from ADA positive subjects should be carefully evaluated. There should be some assessment made whenever you get positive samples and then kind of comparing a little bit to, or not comparing, but evaluating that data with your PK data. There have been instances where regulators have asked for the PK analysis to be performed with and without the ADA positive patients. What impact does that have? How does this change your understanding of the PK analysis or of the particular product? And then lastly, just a couple of word. It's not. I don't think we'll go too much into detail about the NAB assay, but something that we've been seeing kind of developed in the past several years as well. So we're seeing more and more acceptance for NAB assays using the LBA format, using ligand binding assays for the biosimilars. There's a couple of key criteria here, though, to have a successful utilization of the LBAs for NABs. Antibodies without the ADCC and CDC activity would be the most appropriate, and those that are designed against soluble targets. Again, the regulatory focus around this, though, where you will really see the scrutiny come in if you use the LBAs for NABs is the positive control. What is the positive control you're using? Is it the most appropriate? The drug tolerance of the assay, the NAB assay, and the target tolerance. Regarding the positive control, now typically we've been using monoclonal antibodies for our NABs, and you can still continue to do that to demonstrate the sensitivity. But it should also be the same positive control that is used in the screening assay, in the ADA screening, should be used for the sensitivity assessment in the NAB so that you can kind of do that comparison and understand that, and then either one can be used as your system suitability control as you move forward. And now we turn our attention to our biomarkers, or our PD assays. So biomarker assays used in biosimilar development, we typically, this is the relevant section in the bioanalytical guidance with regards to using our biomarker assays. And typically, as with a lot of things, we use our fit for purpose approach. So we can determine what the extent of the method validation is, because biomarkers can have an even more, a wider latitude of applicability. So something to kind of consider, I think it's fairly comprehensive, is the guidance from the Critical Path Institute with regards to the use of with regards to the validation of the assays used for assessing the biomarkers. So biomarker method validation in a snapshot here, we can kind of see that we're typically using the typical parameters that we would see during our PK validations, our accuracy, precision, sensitivity, selectivity, and so on. Now, here is where a lot of times again, we're returning to the use of commercially available kits. Now, with biomarkers, there are a lot more availability or prevalence of commercial diagnostic kits. Now, these are designed for use as clinical diagnostic tools. However, one thing we need to remember is that they were designed for such a use, for clinical diagnosis. It may not be appropriate or suitable for use in drug development. Typically, we need a different type of characterization for example, the calibration curve. Again, a lot of diagnostic type kits might not have a sufficient number of calibration curve points across the calibration range that we need for drug development purposes. And then sometimes some of the QCs provided in these diagnostic kits aren't as well characterized, or the concentration isn't always known. It might be a very, very big range. And again, for our purposes and using it for, biosimilar assessment and studies, we have to be using QC concentrations that are known and established. Typically, the standards and QCs, if they so happen to come in them, in these kits, might not be prepared in the same matrix as the subject sample. So they may be matrix derived, like serum derived or plasma derived, or there might be some proteins, but it isn't an equivalent type of matrix as our samples would be. So this is fairly important to always, because we know that our standards in QC should be in the same matrix as our study sample. The analyte source or the reference standard that's in the kit, it is different typically than what you would need to use. It would be different from what is being given to the subject samples. So for example, it could just be a protein isoform rather than the whole molecule that is being used during the study. So testing or evaluation could be done to evaluate the differences in that during assay performance. But again, it is typically incumbent on us to do that assessment. And then during the life of your validation and sample analysis, it is it's very possible that you will be going across multiple kit lots. So even within a study, it's possible to use multiple kit lots, let alone, across studies. So it is important to understand, if any assessment is being made by the manufacturer for lot to lot, and it's also important to have your own internal procedure for doing lot to lot variability testing or comparability testing. Now, there are other commercial kits that are available and are designated as RU or research use only kits. So some of the other challenges that we have with these commercial kits, there is no industry standard for, standardizing the development of these reagents and designating the quality attributes that we have for these kits. And there has been some kind of industry movement on this or looking into this so that there can be recommendations made to the different stakeholders of how we can standardize that. So there is definitely an understanding between the manufacturers of these kits as well as the end users of these kits. Right now, the lack of the standardized approach makes applying the validation of these kits for using biomarkers going into use in our regulated bioanalysis, it sometimes can make it difficult. But I think that we do need to address that because there will just be more demand for biomarker data, but it has to be quality biomarker data. I think that more and more in the coming decades, as we do more and more of these biomarker PD, we have to have a good understanding of what the different manufacturers are generating and giving us so that we can use it with the most appropriate understanding as we go into our validation. And so it is important to have the requirements so that kit quality can improve and standardization can be implemented across the industry. So one of the things that, like I said, are different types of stakeholders. I have been looking into this, and one of that is what's called the Global CRO Council, or the GCC, and they recently, fairly recently, released recommendations on how we might use a classification system for these different LDA kits that are being used for biomarkers. That is intended for use in drug development. And so this is just a summary of what they have kind of given as recommendations in this paper. So, you know, again, as with a lot of the things, we can take these kits and use it in a fit for purpose manner, right? So in early phases, exploratory work, initial stages, we don't have to have as many things established or characterized, right? If we're doing exploratory validation, there are some things that could be optional. So, for example, reference material throughout the whole system, especially as we go to full validation, it has to be well characterized. But as you go to early development work, some of the parallelism data might not have to be necessary. We don't have to have full characterization of some of the critical reagents. As for the use of surrogate matrix, it might be appropriate for use, especially in the early work, but as opposed to like when we're doing tier 1 analysis or gold analysis, where we're going to be using that data, then we have to follow a more stringent standard. So this is just a snapshot of the different type of uses, different types of kits that could be considered based on the intended use or the context of use that we would do. And again, how we would want to approach that kit development, right? Across the different types of parameters that we always evaluate. So as, for example, precision and accuracy, or our LOD versus our LLOQ, and so on. So the intended use of our end user will determine the type of kits that they would use. And this is the recommendation that has come out of that GCC. So by looking at it, you can kind of get, if it's a bronze level kit, you can have a fairly good understanding in your mind about, okay, what can I use this for, and how well characterized are the reagents of this kit, and how well established is the methodology, versus a gold standard kit where you know that this is definitely, all of the necessary components have been tested, all of the necessary characterization has been done, and that it can be used for, using for appropriate for a full validation where data submission to the FDA or any regulatory body might be considered or in consideration. So what do CROs need from these commercial biomarker kits? Typically because, a lot of times we use RO kits. So as you've seen before, you can utilize different types or levels of development of these kits. And I do think they're all equally necessary. You don't want to waste too much time when you're doing exploratory work, but you definitely want to have a well-characterized and well-established kit by the time you're doing your clinical, your phase 1 data. Something though that kind of goes through across those different types of levels, though, is to having a well-characterized reference material, establishing the lot-to-lot variability and knowing what that is, and the parallelism data. Those are I think the top 3 most desired feature in these kits. And especially the lot-to-lot testing to minimize that variability, because presumably or conceivably this can go all the way from your exploratory work all the way to your phase 1, phase 2 type study. So a little bit digging into our reference standards that we were just talking about. The change in the reference material law, right? So typically this would apply to our biological drugs, but sometimes logistics is kind of a necessary headache that we have. We might not have this exact lot that is going to be intended to use in the study. We might not even have the specific production mode that is being used for the study or will be used. So it's always necessary to keep in mind that a bridging might be necessary when that study lot is available. You have to do at least 1 run. We recommend 3 comparison runs to ensure that it is repeatable and that it's there. But it is something that you have to kind of make sure that is well established in that changes in your lots is not going to affect any of your assay parameters or your assay performance. So, here's just kind of an illustration, right? This is the reference material, because this is the heart of how you get accuracy in your results, in validation and across time, across your studies. So the guidance does tell us we have to appropriately characterize that material. It would be good to kind of assess the different sources of these reference materials. However, the gold standard of reference standards, whenever it's available, is the NIDST WHO type of standards. And you can see here the different sources here where we've compared the different type of standards for this particular assay. And then one thing to keep in mind, and again, this is another thing that is like kind of more and more coming up, especially as you go across studies or studies that go for a fairly long time, right? Is the trending of your QCs. Now, appropriate, here, this is kind of an example of the trending module from our AGRIS software, where you can do across studies. This happens to be doing this across comparison across studies. So you can kind of see like how this particular assay has performed through its QCs over time. And then as appropriate, you can internally set up your criteria for how these will be assessed. So a lot of people have looked into using either the Westgard rules, typically used in clinical laboratories, but being adapted for use in our bioanalytical laboratories. Now, the reference standards that we get from kits, should we be using that in our PK assays or our biomarker assays? You can use it, but we do recommend to not do that. Again, while it is feasible with the appropriate justification, right? I do think that it will, in the end, cause you to do a lot of work, because you do have to do a lot more comparison work. So while it could be interesting, it might not be necessary. So appropriate justification, you have to compare your kit calibrator with your reference product and your biosimilar. You have to do it, minimum of 3 assays. But then what does that mean when your lot changes? What if there is a systematic bias, even though it meets system criteria? What is the stability? How does that play into the stability? So I do think that this is something to kind of evaluate. I mean, if you're doing initial evaluation, certainly, again, we kind of always keep in mind, right, the context of use. But in general, especially as you're going into your clinical studies, it is incumbent that we use the highest standard, or guide the highest standard as much as possible in doing our validation. So lastly, for all the things that we kind of talked about, we have, I've touched about it here and there as we've gone through our PK, ADA, and biomarker assays, is the software system. So this is another thing that's kind of gone leaps and bounds in the development and advancement in the last several years. But because of its more widespread use and implementation in our laboratories, we've also seen significant scrutiny on these electronic systems during audits. So in general, I find that there would be at least one auditor focused on the study data. There would be at least one auditor focused on the electronic systems, sometimes even more if you have extensive or multiple electronic systems. So one of the key things that kind of always keyed in is digging into your audit trails. So it kind of is incumbent on you. You have to know all of your audit trails. You want to know more about your electronic systems than the regulators do. You want to know the ins and outs of your system. You want to know the ins and outs of your audit trail. You don't want to find out the existence of any facet of it from your auditor. And at the heart of that, I think the outgrowth of that is one that I think most people are familiar with, is the US FDA 21 CFR Part 11. So some of the key features, what does it mean? What does it mean to really comply with 21 CFR Part 11? So these are some of the key features, but it comes down to how can we best say preserve and the integrity of the data that we are generating, and to ensure that it does not get any tampering. And so that's where our audit trail comes in. And we have the ability to save our documents in read-only format. So that's where our archiving or our archivable abilities come in. So what are these key features that kind of some of the things that I think are rather important with these electronic systems? So you have to just automate the audit trail. It can't be something you have to generate. Otherwise, compliance is always going to be a big question of that. The security is of paramount importance. You can't edit anything. You shouldn't be able to edit anything. And if it does allow you to edit, it has to capture what you're editing, why you're editing it, and who is doing it. Just like we would for any of our paper-based systems. And then that it has to be archivable, meaning that once it's locked, archived, whatever terminology we want to use, it can't be edited. You have limited access. Only appropriate personnel should have access to it. And when needed, limited user access or read-only should be available. So, for example, if an auditor comes in, we have to have the ability to retrieve that data and show the data. So, in summary, for how we move forward, given all our more clarified and more rigorous standards and guidances that we've gotten over the years, as well as in hand, the way our technology has advanced and all the enhancements that we've kind of experienced in our technology across the different platforms that we're using. First and foremost, a robust PK assay, you kind of need it. It is at the heart of how we would demonstrate that biosimilarity. Method development and validation, you require reagents that have been well characterized. And the use of statistical approaches, very robust statistical approaches in doing our immunogenicity cut points across the board is definitely required. Using the use of kits across different types of utilities or different types of approaches, it's always incumbent on us to always be very well defined on the context of use and how we're going to be able to use that data, generate that data at the end. And then finally, understanding our software systems. You have to know the ins and outs and how if there will always be some incongruities or weakness in your system. You have to know about it, and you have to know how to deal with it and address it head on. So having said that, I will wrap that up, and I believe we go to Q&A.
Thank you, Clarinda, for such a wonderful talk. It covered PK, LBA, biomarker.
Yes, I do realize I've taken you. I've taken you through the entire landscape quite fast.
Exactly. I think in the interest of time, there are a couple of questions, if you can quickly answer them.
Yeah, Patanjali, just before that, Clarinda, if you can stop sharing so we can project your video. Yeah, exactly. Thank you. Okay. Go ahead, Patanjali, please.
Yeah, thank you. Yeah, I think I might, it's a virtual conference, so you might hear a virtual applause. You can feel the virtual applause, if I may say,
Well, thank you.
Yeah. There was a question from Srikant Dettakavi. I hope I pronounced his last name correctly. When we use surrogate matrix for endogenous compounds in LC-MS assay, the internal standard variation will be observed between samples and the CCs and the QCs. Any thoughts on how to address such issues?
So we've actually had to address it a couple of different ways, and it actually is. I wish I could give you a magic answer for one size fits all. It's very, very dependent on your molecule. So for example, one where we've actually approached it both ways. I will use the teriparatide example that I used earlier. Sometimes for the particular dosage that you're using, it is more appropriate to use the LBA approach. It's well established, it's well characterized, it's something that we've been able to use across multiple studies. There was one particular one where we had to actually use, or I think it was actually a sponsor request, where they did want to use LCMS approach or methodology for doing the teriparatide. And here we know, right, there is going to be some endogenous interference in there. So really, it's going to come down to what I had kind of said before, in a couple of different ways that we optimize that assay. One is the use of the surrogate matrix was necessary. We had to kind of just show the justification of that in the development, and then it is critically necessary to have both types of QCs that you use, that you demonstrate in validation and that you continue to use in your sample analysis, meaning your QCs that you do in your surrogate as well as in the matrix, basically.
To demonstrate the parallelism.
Yes, exactly. Okay.
There's another question in the Q&A process. How dilution linearity is performed? What are the different samples analyzed during this assay?
Dilutional linearity?
Yeah. Oh, how is it performed?
So if I understood correctly, what types of samples? So it's typically our contrived samples or our spiked samples, right? And we just want to basically demonstrate the linearity across our range of the calibration curve. And two, typically you kind of already can calculate how far or how high your dilution will be required during the study, by knowing what the dosing regimen will be, as well as using that as well as the Cmax to calculate where your PK samples will fall. So using that knowledge, you can estimate where your dilutions need to fall, and then make sure that you demonstrate that during validation.
Yeah, I had several questions, but in the interest of time, I will pause here. The question I had was, any idea of false positive rate calculations during your AD assays, FPRs, during the biosimilar analysis?
So, yeah, I think if I understood what should be our, how do we do that? One of that case study that I was using right there, where we kind of ran into trouble with that is because I do think that we did set that cut point a little too low. Meaning we were really pushing that, right? But in turn, we were, there is a possibility that you will get your false negatives, right? Yeah. So in essence, it is always recommended, it's actually recommended that we design these methods to have about a 5% false positive rate go towards the false positive, because as it lucked out in our study, it was deemed, when we had those additional positive samples, it was deemed that there was very small possibility, or none at all really, that those would have converted or confirmed positive, right? So, but still, that perfectly illustrates the fact that it's much more preferable to have those additional positives in screening rather than finding out later, especially as you do your in-study cut point. This is also the other importance, or the other side of the coin, of the in-study cut point. That is where that other piece of importance comes in, because originally, I don't know if I mentioned it during the initial discussion, originally the validation cut point was used, which is why things, those 5 samples were below the cut point. But when in study was used, it was demonstrated that there were 5 additional ones. Now further justification and additional testing was made to show that, hey, but these were never going to confirm positive. So
in the interest of
rather than get through that whole thing experience, it's probably better for your heart rate not to go through that experience.
In the interest of time, it's a good segue. I think we're going to invite you next year for doing the presentation on cut points and things like that. Hopefully you're available.
And hopefully I will be there in person so that we can, we can demonstrate that all together, right? In person.
And it's a nice segue. Deepak, go ahead and present the appreciation certificate. Go ahead, Deepak.
Yes, so would like to
We lost you. Are you there?
Oh, did we lose?
Okay,
post
anyway.
midnight for Clarinda. So
Yeah.
we really appreciate Clarinda for a very great talk you gave. It was very informative, very elaborative. I think you covered so many things within this. So thank you, and we wish to have you in person when we do next year APA India 2022. So thank you, Clarinda, for your presentation and for your time, for staying awake so late in the night. Thank you, Clarinda.
And thank you for inviting me. Thank you to the organizers for inviting us and allowing us to give us this talk.
Thank you. So now, may I now request our moderator, Shefali, to introduce our next speaker.
Yeah. Good morning, Deepak. And I have the pleasure today to introduce Mr. Amarnath Jaiswal, speaking on bioanalytical challenges, a new learning, which is in keeping with the monthly theme of regulatory challenges, learning, and opportunities. Currently, Amarnath also, he's been a very active member with the APA, helping us, enlightening us every year with amazing talks for now about a decade. And currently he holds the position of Vice President and Head of the Bioanalytical Department and Complex Molecule Analytics at Viatris, India. He received his MSc Tech in Pharmaceutical Chemistry from University of Lucknow and PhD degree yet to be awarded in the coming days. He started his career with Sun Pharma research labs in the PK department as a bioanalytical scientist, and gradually has grown up to a position to lead a team of scientists who are involved in preclinical PK, complex molecule characterizations, in vitro and in vivo BE studies. He has been scientific, knowledge guide, has leadership skills and hands-on experience in conducting human bioequivalent studies, in vitro bioequivalent studies, characterization of complex large molecule analysis, and experience on handling extractable, leachable studies and biochemical assays, as well as using various analytical methodologies such as UPLC, LCMS-MS, GPC, ELISA, ICP-MS, Zetasizer, FPLC, and QTOF. So it is my pleasure right now to invite Mr. Amarnath Jaiswal for his enlightening talk. Thank you.
Thank you, Shefali, for giving us such a wonderful introduction. Let me share my screen, and one can confirm once it is visible. Can anyone confirm if my slide is visible?
Not yet.
Just a minute. Okay.
Yes, it is.
Okay.
Yeah.
Okay, so thank you, Shefali. Thanks, Deepak. And I have been with APA, it's almost 8 years. And if you recall, I used to give a lot of, the talks related to the regulatory, the observation, regulatory comments, the learning which we had. And since we are in a pharma industry, we closely work with a lot of CRO, not only in India, but globally as well. So prior to this talk, I used to talk most of the time on the regulatory comments and the observation and how those the regulatory query can be implemented into the procedure so that those the regulatory query can be avoided. But this time, what we thought that rather you discussing on the regulatory query, let's look at what are the, the bioanalytical scientists face and the challenges when they are developing a method, or when they develop a method, right? How truly they can believe that, their method would work perfectly. The SA method which they are going to employ on, the clinical sample analysis or during the validation, what are the challenges they faces? So I thought that let me give us some respect to the bioanalytical scientists who work for us, who work for a organization day in, day out, and trying to develop a robust and accurate method. But there are occasions when those as a method, even it's a fully validated, doesn't work when it comes to a in vivo sample analysis. Or sometime when it works very well during the study sample analysis, the quite often we have seen that it fails during the in-court sample reanalysis. So there are certain challenges while developing an SM method, validating them, during, the apply and the study sample analysis are during ISR. So what we thought that let's take some of the case studies where we have seen that, the essay fails to demonstrate the accurate and reproducible result. And that was my the talk today. Let me go through a couple of points. So this is just a disclaimer. Uh, some of the data which is from my organizations, but those are not so confidential, and that's the reason before the giving those data, I need to just give a disclaimer. Okay, so I'm going to talk today about four of the drug products where the method has been developed and somehow it got failures during the conduct of the study sample analysis. Somewhere we have found that there was some issues during the sample handling at the clinical site as well. And there are certain challenges during the method development itself, and that allow the bioanalytical scientists to look and work out of box thinking. And probably that is the unique approach which we have seen it. And that's the reason I thought that let me share those unique approaches as well while somebody is in developing a method for a very, very low detection limits. I mean, that was also the good learning for us. And that's the reason I thought that let me take those case study as well. That is a respiratory drug product. So first talk would be based on one of the injectable products called as iron dextone. I mean, majority of I have seen that a lot of the CROs, even in India, they are working on iron complex product, and there are certain challenges in the iron complex product. So some of the information may not be reflected here because of the critical parameter, but at least they would have some idea that what kind of the difficulty people are facing it, and how those difficulty can be minimized when it comes for a study sample analysis. The another product is the diltiazums, where we can go and discuss. There is a different kind of the issues which we have, the identify what the issues are. The third case is related to the rivaroxaban. This is a COPD drug product. The another is a ferric carboxymaltose. Again, it's iron complex product. So except the diltiazem, rest all the three studies, the patient study on a multi-site, almost 20 and 15, the clinical site has been involved, and there is there really a lot of challenges in terms of the recruiting the patient and dosing the drug product, getting the sample, and what are the challenges being faced at the clinical site as well. So let me look at, the journey of a bioanalytical scientist. I mean, it's not so easy for a bioanalytical scientist when it comes to develop a method. When somebody says that, we have the successful method, SA method is available, we cannot say truly that your message is really a successful until it is not fully validated. Sometimes your method gets validated, there is no issue at all. When it comes to the in vivo analysis, your method may not work. And even though your study data looks like fine, there is no failure rate, everything is fine, but there are cases where people used to, there is the data where we have seen that they fails to demonstrate in core sample analysis. So when a bioanalytical scientist say that my method is, the robust, rugged, and accurate, until it is not demonstrated your in-course sample analysis, still it it is not the correct to say that my SM method is completely, the demonstrated accurate and reliable data. That's the reason at every moment of time, when it comes to the assay validation and view analysis and cut sample analysis, there is always a possibility that your assay method may not be 100% give a reliable and accurate result. There may be the point where your batch gate fails. You may have a lot of incident investigation as well. So again, depending on the number of incident investigation, that also reflect the assay validity, your assay accuracy, reliability of data as well. So that's the reason I thought that no, let me also give a respect to the scientists who work in the lab, and really a challenge for them until their methods developed by them is not being demonstrated with the 100% or 90%, 85% of in-core sample analysis validity. Okay, so when a new product is given to a scientist, whether it's a generator product or it's a new molecules, the first product is R&D extend, where I'd give you some of the information that it was really a very challenging, because there was no PK data. There was no, the prior PK study was carried out. So when it comes to a new product to a biomedical scientist, what potentially they used to look at, the task for them is to find out the right calibration curve. To find the right calibration curve, they also need to look at what a type of formulation is. It's a oral solid dosage, it's injectable, it's a respiratory products, it's a biologics. I mean, these are the certain points they always need to keep in the mind, that what the type of product they have, what are the various dosage strength they have, whether the study is going to be done with the normal healthy volunteers, or it's going to have the patient study, because that also critical aspect to look at. If it is going to be a patient study, how the outcome result would be, what would be the Cmax range would be. Again, there will be various doses form as discussed. The another challenge for them is how to select the right internal standard is, what is the isotopic enrichment with the internal standards is. Nowadays, you probably, the people who are there in the lab, they face a lot of challenges. They used to find a lot of interferences at the analyte to retention time, because if it is not the 98%, 99%, 100% enrichment, they would end up with a certain problem. Again, the purity and authentication of the compound is very important. If it's your in-house compounds, probably you may not end up with the problem. But if it is a commercially sourced, probably that's very important. Someone to look at all the characterization data to make sure that the compound they are going to use for their curve, and indeed they are going to generate a in vivo study data is considered to be reliable. Otherwise, tomorrow they will end up with the problem if their data is not going to meet with the reported literature is. When they develop a method and apply for for method validations, again a challenge is for them how their run performance is, what is the number of failure is, how the instrument performance looks like is. If there are a number of failures, probably that method may not work truly when it comes to the in vivo analysis is. And I said the in vivo analysis, again, your run performance given a reflection about the assay performance is, what the number of failures is, how your I study response in the curve QCs versus subject sample is. I mean, many of the times we have been witnessed that agency always come with and ask that, I study response is something else in your control sample, and that's showing a different into the study samples, right? And that's the reason in the last year, even the FDA has released a guidance on that and given a lot of example that if there is a significant difference in your curve and the QCs versus a static sample, probably someone has to look at and given a justification how one should go and accept your data in terms of considering the reliable values. Again, the very important is the Cmax which you observe from your study sample, how close with the reported value is. And majority of times we have seen that, the bioanalytical scientist doesn't focus on those part. They always focus on how my curve performance is, how my QCs is, how my failure is. But at the end, when the agency comes and they try to look at how the reported data is, and if your data is not close to your reported value, again, it would be a question mark. And then it started looking, the lot of investigation, and somebody may find that, my standard was not fine. There was something wrong with my assay method. So probably this is also one of the aspect that, when the bioanalytical scientists working on any kind of the study, they should also look at that what the reported value is. And when they generate the data, they should also look at how close the data which is generated in the lab is close to the, the generated product is. Sometimes they may also be witness and they found that there is, the additional peaks into the chromatographic run is. So it's not like that, your data is coming fine. We should completely ignore that my additional peak chromatogram is, but it's equally important that someone has to go and look at what this additional peak is, how important is, if something is get degraded or something in additional peak, which is also going to impact my outcome result is. And there are cases where, people used to get an abnormal value. So majority of time we have seen that across the lab, they had a certain procedure to identify how they can identify abnormal value is. They also define a certain procedure, how they can identify, how they can analyze them, how they can say that this abnormal value is not because of the potentially due to the bioanalytical assay, but that can be a clinical issues, or there could be a pharmacological behavior of the subject. But something need to be found, and the to before submission of the dossier, they need to have a robust investigation procedure to determine and the control and you know confirm that the abnormal value is not because of the potential concern from the bioanalytical or the clinical aspect is. And then of course the ISR performance and reproducibility is one of the the task that everybody has to look at. So let me see, the first case study is the iron dextran. This is, the complex ferric oxyhydrate hydroxide molecule, which is basically the iron, which is coated with the dextran or the sugar, which is the molecular weight is around 5,000 to 7,000 is. And this is a parenteral preparation is, which is potentially used for the anemic patient where the OSD therapy is failure, and then the doctor suggests to go for the injectable therapy. So this is a kind of the product which is that really help us for an anemic patient. The study was conducted the almost a 20 clinical site on the anemic patient. And as per the guidance, it was recommended to estimate a total serum iron as well as a transferrin bound iron. Unfortunately, there was no PK data available because that's the first to file project is. So that's the reason it was very difficult to find what would be the Cmax ranges. So that was the first the issues from the bioanalytical scientists, how they would come up with identify the calibration ranges. And since there was no intra-subject variability data was available, it was really again challenges. So somehow we have a collected couple of literature, and then in past we did a couple of other iron complex products. So based on those, we have come up with certain the curve raise, their LLOQ and ULOQ. But the challenge in this study was when we have started analysis, we realized that the transfer in bound iron concentration is very high, which is unexpected. And there was a certain patient where we have seen that the sample looks like a swap from one patient to another patient. So let me go and show you so these are a couple of, the literature we have collected, how the transfer in bound iron data in the healthy subject as well as the patient study is. And since there is the limitation in the transferrin protein and in the body, in the serum, that we realize that, in none of the cases, the iron which is bound with the transferrin protein would not exceed with the more than a single disease. Even if you look at the total iron binding capacity in the serum sample, either in the healthy subject or even the various the patient which is suffering with a lot of the disease, including the anemic one, it should not exceed with the 4.5. But based on the previous couple of study, for example, in the iron sucrose with 100 mg of the injectable drug product, though it was in a healthy volunteer, there was a patient study with the 750 mg of the doses formulation. We could see, and then there is another ferrimoxetol. Again, it's the iron complex product with the injectable product on a healthy volunteer. We could see that in none of the cases we have seen that it exceeded more than a single digit. So based on those, we thought that let's go and, the set up your curve range with 0.54 to, the 12 microgram. And since the total iron, we have the data from the iron sucrose, and we have seen that, for the 100 mg dose, it has not increased more than even the 30 microgram. And since this is a 50 mg dose, let's keep only with the 15 microgram. And then we started, and I'll just give you the couple of, the workflow, how the transferrin bound iron, the SM method is being set up is. So typically in any of the lab, this is, the typical workflow for estimation of the transferrin bound iron. I would not discuss in the total iron because it's very, very simple SM method, just in the hydrolysis with the nitric acid. And you can just break all the iron present into the body is. But transferrin bound iron is always a challenging, the aspect is. So there are a couple of the places which we have a mark in a red color. That is a very, very, the critical parameter which is really a contribute in terms of getting your accurate and reliable data. For example, the volume of the serum, how much the volume of serum is going to be taken. Let me. So the volume of the serum is very, very important. The phosphate buffer saline, the volume is also equally important, how you are going to dilute it. And then when the selection of the solid phase extraction cartridges, we have a couple of data. We have used almost variety of the solid phase extraction cartridges, and we show you some of the data now. And during the method development and even in the method validation, we have demonstrated the solid phase extraction with the amino, the chemistry with the 1 gram bed capacities gives a very consistent, the accurate result. The centrifugation is speed, the loading of the samples, how many times you are going to load it, and then the fraction, then further dilutions within 1% of 90 cases. So this rate parameter is very, very critical, and it's always a task for a biomedical scientist to optimize the best way to get, the reliable, the transferrin bound iron during conduct of the studies. Okay. So a successful method development and validations was performed. And this has been demonstrated with, the use of the free iron and with the iron dextran product as well. And we realized that there is no issue at all. Let me remove the pointers. Okay. So when we have initially started the analysis, we thought that let's analyze with a couple of subjects in the first goal, because it's a big study, it's almost more than 300 patients. So initially we just analyzed a couple of subjects. I just wanted to give you some of the representative data. Now, when we have analyzed the subject, we realize that the transferrin bound iron concentration is very, very high in a couple of subjects. Ideally, it should be a single digit, less than a 10 microgram concentration. But when you looked at some of the subjects, like here you can see, here you can see it. And then again, in another subject, probably you can look it here. Anyhow, the transferrin bound iron concentration is very, very high, and it looks like the profile is very good. So one cannot say that there is an abnormal value, because the PK profile at 1.25 years to 60 nights looks like a very, the absorption phase, then it's an elimination phase. And then if you look at the another subject, I mean, in a first intense, it looked like the sample get swapped from, 2.5 hours from one subject with the other subject. And similarly here, you can probably look at, and it's a clear-cut indication that there would be some sample swapping. It can be at the clinical site, it can be at the bioanalytical site as well. We can't say, and we can't ignore that it would not happen at the bioanalytical site. But the point to be noted here that the elevated concentration, which cannot be, the reliable data is. And since there was no PK data available, we thought that let's stop the analysis at this moment, and let's, evaluate it, what could be the reason is. So in this study, we found that there are two potential concerns. One is the overestimation of the transferrin bound iron data. Another is a clinical sample swapping is that, and that's the reason we have stopped the analysis. And then we started the investigation is. When we started looking, the investigation, what we thought that let's look at what are the various type of the iron present in the serum is. When we have extensively looked at the literature, we realize that there are potentially 4 types of the iron available in the serum is. One is the free iron, The another is the heme protein, another is the albumin protein, and the non-heme protein. Typically, the free iron is very, very low concentration in the serum, because if the iron free component is increasing to the body, that leads to the cytotoxic effect, and probably the patient would not live. And that's the reason the free iron concentration would not be a more than a zero 0.1 microgram is. So we realized that, the free iron would not at all there. Then we also looked at what could be the heme protein is. We realized that the hemoglobin, but the hemoglobin, the iron associated with hemoglobin would not be the freely available in the serum. So that part also get eliminated. We also looked at that there is an albumin protein, which is a non-specific binding site. So let's look at the albumin protein. With the iron bound with the albumin protein, probably it would not be retained on the SP cartridge, and it is being passed through from the cartridges, and that's potentially giving, the elevated concentration of transferrin bound iron is. The another part is the non-heme protein, which again categorizes in transprotein, lactoferritin, and the ferritin. So transferrin, as we already discussed, that should not be increased more than a single digit. Lactoferritin, probably it's available into the milk and the saliva, so that is also ruled out. The ferritin is actually the reservoir protein, and the concentration of ferritin again it's not very high. We could see that it's not more than the 0.27 microgram per ml. So that we have eliminated. So what we thought that let's look at the first why there is a sample sweeping, whether it is as a bioanalytical site or it's as a clinical site, and then there are another thought processes in terms of getting the elevated concentration is. Like the iron dextran, if it is an unchanged form into the serum, which is not being retained on the cartridge, there could be, further dissociation of iron dextran project into the body, and that gives a lot of the free iron. But as I said, that the free iron would not be freely available in the serum. There is another, the part that, the other protein, like we discussed with the albumin. The solid phase extraction cartridges, the chemistry which we have utilized for the study may not be suitable. The amount of material which is filled with the solid phase extraction which is not suitable. And then the volume of the phosphate buffer saline that can potentially impact is the serum volume, the dilution volume, and then the centrifugation speed, because that the different RPM may also potentially impact on the outcome result is. So in the first goal, what we have done it, we have just taken a couple of subject sample along with the control samples and just reanalyze with the original assay method to see if the data is reproducible or the profile data is remain same. If you look at this is an original value, this is investigation, the data. If you look at after the high concentration value, there is a low concentration value. When we have repeated these samples, we again found the similar kind of the profile that indicates that there would not be any concern with the bioanalytical side or the clinical aspect, because the data is remain same, the profile still looks like the same. But the potential concern this time, there is no reproducibility of the data here. You could see that the percentage difference from the original value to the investigated data is quite high. The point here is still we can't say that my assay method is perfectly fine. So in the first instance, we realized that this assay method is not going to work at all. Because initially when we have analyzed it, the value was 25 become a 35. Again, this is in a higher concentration. When we look at the curve and the QC performance, it was perfectly fine. The area counts was remain same. There was no change. I mean, potentially the first time, if the people looks it, they probably thought that, the area counts will increase, the curve impact and all, but we have looked at there is no change at all. The instrument performance is remain same is. Okay, so then what we thought, let's dilute the sample, because it's still this data which we initially analyze it, it was out of the range. And when we have seen that, because the curve range is only 12 microgram and the initial value is 20 and 25, which is out of the range, let's dilute the sample and analyze them. So what we have done it, we have taken the sample, the different subject at different time points. We have taken a subject as such, and potentially the assay method requires a 200 microliter of the serum. We have taken those sample again with the 1/2 dilution, and the same sample we have taken a 1/4 dilution. And you could see here, when we have taken the sample with as such, again, there was a concern in the outcome result. And At the same time, it was a surprise for us when we have a diluted this sample. Let's say when we have a diluted this sample, uh, this one 25 and 20, it become a 5 and 3. When we have taken this sample again, 25, 3.6, let's focus on only one sample, one for dilution. Again, this data is altogether different. So that also indicate that my dilution factor, which was fully validated during conduct of the validation, is not going to work because after the dilution, the data is not again aligned with the original value. At the same time, when we have diluted our control sample, you could see the differences between the initial value and the repeated value is very, very close. It is almost close to the 100%. So one thing it was clear that assay method is working absolutely fine when it applied to the control sample. When it applied to the subject sample, it is not fully demonstrated in terms of whether you are going to take the same elocution volume or you are going to dilute it. So that trigger that is then we need to work, on the assay method, and then this method is anyhow not going to work at all. So we have hold any kind of the clinical investigation because still we have started, the questioning our assay method itself. So let's see here. What we have done is we have taken a blank serum. We have co-spiked with the API product, not the drug product, but the API we have taken with the different concentration. If you recall, initially we have demonstrated our assay method with 20 microgram per ml. So we have just increases the API concentration into the serum, 20 microgram, 30 microgram, and the 40 microgram. Because initially we have seen that the concentration did not exceed more than even the 30 microgram is. So we have taken the highest concentration of 30 microgram, and then we have spiked and trying to look at what is the further pass-through concentration of the iron at the bottom in the fraction. And we could see the contribution of iron from the API is only 0.5 microgram. Even when we have a spike with the injection, we could see that the contribution is only 1.1 microgram, which is again, it's not correlated with the actually subject data. Then what we have done it, again, we have spiked the free iron standard up to a 60 microgram. And we have seen that the contribution is only 6.6 microgram. But the question here is, in the body, the free iron would not be more than even the 0.1 microgram is. Though the free would not be able to retain on the cartridges and the contribution is still 6.6 microgram, that again, we concluded that this would not be in a case. Still, we are able to retain 89% of the iron onto the cartridge surface. Then what we have done it, again, we have taken, the human serum albumin. So because we thought that maybe the albumin, the iron which is bound with the albumin can not be retained on the cartridges and that is being passed. We have spiked the very high concentration of the free iron up to 60 microgram. And at the same time, we have also spiked the test product and the RLD product up to a 40 microgram to see what is the loading capacity of our cartridges itself. And we realized that the contribution from the serum albumin, when it co-spiked with the iron 60 microgram, is giving only 1.4 microgram and 0.7 microgram. So that would not be a case with the actual study sample is. Then we have looked at the different solid phase extraction cartridges with the different bed size, the 100 mg bed size, 500 mg bed size. And this is one we have used into our assay method, 1 gram capacity. We have also taken with the different, the brand with the 1 grams. Then again, we have taken the polyamine with the 500 mg. And probably we could see that, the contribution with the 100 mg is giving almost, the 21 microgram. But since we are not using these cartridges, that has been ruled out. Here we have ruled out. And here the contribution, which is the actually used in RSA method, is just 0.3 microgram. So that reflects that the cartridges which we have selected in RSA method is perfectly chosen for the assay. But unfortunately, this is not giving the right result which we are expecting it. We have also looked at the cartridges which we have seen that there is the uneven the bed size packing is. In fact, we have also seen that there's a lot of the air space. There is a free flowing the cartridge into the into the cartridge. This stationary phase looks like a free flowing. So we thought that probably that could be one of the reason that is not giving the consistent results. And that could be, the potentially one of the reason the iron is not 100% retaining and the cartridges is. What we have done it, we have taken the different lots number. We have taken the free flowing cartridges. We have taken the cartridges where we have found that there is an air gap with the different one. We have also taken a couple of cartridges which was open and kept in the lab for several days. But if you look at the control sample with the accuracy, which is very well acceptable, the accuracy is almost within 85 to 115. We have not seen any kind of the abnormal value. So what we realized that this phenomena would not be a true in case where we used to get an abnormal value with the study. What are the other trials we have taken it? We have try with the different phosphate buffer saline, the serum volume. Instead of the phosphate buffer saline, we have also used the water, the different RPM we have looked at, the different, the time interval we have given, and across all these, none of them we found anything which can be correlated with the data which we have obtained. I mean, this took almost a couple of months time to spend in the lab, and it was really challenging for us to find out what is the real data is. Probably the real data is the elevated concentration. But tomorrow, if the agency will submit this dossier, agency may not accept because in a true sense, the transferring protein itself is not very high in the serum. So that was very, very challenging for a scientist to, find out what would be the real data is. Then it was thought that let's try with the two cartridges instead of using a single SP cartridge. Why we can't, the pass through the sample, again, take those sample, again, you put onto the different cartridges. And that was a really breakthrough for us. When we have analyzed with even the very, very high concentration, which was not expected in the study, we have a spike with the 80 microgram of the drug product. And when we have a spike, you could see that the contribution of the high concentration of the iron dextran is almost a 50 microgram. That can give us some sense and correlate what we have observed into the study is. And the contribute accuracy, you can look at almost a 600% accuracy. The contribution of the iron from the drug product itself is a 13 microgram. What we have done it, we have collected this sample. Again, we have passed this sample onto another fresh cartridges. And now you can see the concentration, which initially we found is a 15 microgram, that gave a result is a 2.7 microgram is. The accuracy is within, the 85 to 115. And the contribution with the very, very high concentration of the drug product was only a 0.4 microgram. And that was the breakthrough. And that was, I would say that this was the out of box thinking that, the people have thought that let's try with another cartridge, though it's very, very expensive work. There is a time consuming process, but at least we could able to find the actually root causes. So again, the challenges is not over because this is all a control sample. And we have seen in the past, the control sample, it was working fine. What we did it, we have taken again the subject sample. We have taken a subject sample with the actual volume, which was potentially used in the validation, the 1/2 dilution, 1/4 dilution. And when we have diluted this 23 microgram, we could see the concentration. When we have passed through two different cartridges, we could see the concentration is 3 microgram. When we have diluted with 1/2 and multiply with the 2 factor, the concentration is 3. When we have diluted 1/4 and multiplication with the factor 4, the concentration is remain same. So that gave a confidence that even there is a dilution of the actual study sample, the outcome result is very, very consistent. We again try with another subject sample and all the data is very, very close. And we could see also when we have taken the control sample, that the diluted QC, the accuracy is very, very, consistent, close to the 100%. So that gave her, the confidence that we need to rework on the SM method. It is very, very, the extensive process. It involves almost a 30 the processing steps, but finally we could able to, the fix the problem. You could see this is the initial data where we have seen, there is a sample sweeping, and probably if you put this value here, your data profile is very, very fine. When we have again reanalyzed this sample with the new SM method, you could see all this data is very, very consistent, and that eliminated that there is no sample sweeping either at the clinical site or the medical site. So that was a kind of, the investigation we have done it. And finally, we have, that finalized the SM method with, though it's required a lot of time to spend it, But the key learning from this study was whenever somebody is working on an iron complex product, let's utilize with the very high concentration of the drug product to evaluate what is your loading capacity of your solid phase extraction bed sizes. And probably your SM method may require the very, very high bed capacity. You may require a 2 cartridges, you may require a 3 cartridges to get very accurate and robust results. The another part is what we quite looked at. Whenever people realize that there is the concentration exceeded from the ULOQ concentration, in a first goal they immediately increase their ULOQ concentration is, which is not the right path to do it. Always they need to look at whether those data is really accurate or not. So always, instead of the moving forward to just work on your curve range, let's look at and investigate whether this data is accurate or reliable. The another, I mean, we have just the proven that the sample sweeping is always not at the clinical site. That can be potentially due to your SA method as well. So always it's very easy to figure out that the sample got swept at the clinical site, but always we should also look at if your SA method is not, the accurately give the good result is. Let's come to the another, the case history. This is the diltiazem, which is a very, very unique, the experience we have. This is the normal healthy voluntary study with 120 mg. And the initially, the OZD guidelines released in February 2019, and they state that you need to estimate the parent compound along with the two of the metabolite, desacetyl-diltiazem and desmethyl-diltiazem. And typically, in my experience, when it given in the OZD guidelines, generally the biomedical scientists look at what is the requirement and they try to focus on those aspects which is actually needed for the study conductees, right? And the clinical protocol is signed off in 2019 after release of the guidelines, and the study executed in the last year because of the COVID and all. And the study is, the conducted very well. The analysis is started. So this is just a generic drug product. We know that what is the CMAX reported is. This is the, the curve variance we have selected. I just marked it with the red color because the problem with this assay was with desmethyl-diltiazem. Otherwise, these two analytes, there is no absolutely concern it. The method is developed, validated fantastically. There was no issue at all during the conduct of the validation. When we have analyzed couple of subjects initially, we realize, the desmethyl-diltiazem concentration is very, very high. If you look at it, the reported value is around 21 nanogram plus minus 4 nanograms. So we expected that the concentration would not be more than 30 nanogram is, right? But when we have looked at the initial couple of subjects, we could see the concentration is very, very high. That is not expected at all. Even some of the concentration, the 69, 70, we have stopped the analysis here itself and thought that, no, let's look at why the concentration in this particular metabolite is very, very high, whereas in a parent compound is absolutely fine. The another metabolite concentration is absolutely fine. So there could be a reason. Let's look at what we have done it. And we have a set up the curve range is 0.15 to 45, and we have just highlighted that these are the concentration which is out of the ranges. So it is also not good that, the majority of subject, if the concentration exceeded, you just go and reanalyze them with the dilution factor. The agency may not accept such kind of, the procedures. They would always expect that, the concentration should be within the curve range. There be occasional sample that may exceed it, but not majority of samples should go beyond your curve ranges. Okay, so in the first steps, what typically the lab used to do it, they start looking whether my stock solution is perfectly made it, my working solution is fine, whether there is some error is occurred. That's the reason my outcome result is something different. If there is an error with the plasma spiking, they have also looked at the different standards supplied by the various, the supplier. There are 4 or 5 suppliers. They have also looked at the purity because that can potentially impact your outcome result. They also looked at what the back conversion is, if there is any back conversion from the analyte to metabolite is, or from other metabolite to this metabolite is, because there are instances where we have the witness that the metabolite concentration exceeded or reduces because of the back conversion to the plasma sample. So that took a couple of weeks time to evaluate, but finally we concluded that there is no concern about this aspect is. So what we have done it, we have taken a couple of subject sample representative from subject 1, 6, 11. Here you can see the subject from the various, the sample from the various subject. What we have done it, we have taken the sample, analyzed them as such. We have also taken the sample and diluted them 5 folds and 10 folds, because if you look at the concentration here, the concentration was very high. So what we thought that let's dilute them with the 5 folds and 10 folds, having the experience on the iron extant, let's dilute them and look at the data outcome is. When we have diluted the samples, we could see that there are a couple of sample has some differences, but there are instances where the data is still within, the 20% of the differences. Because this is a single assay method, we could able to generate the second metabolite and the analyte data all together in a single assay method. We have seen there is no much difference in the analyte concentration with the dilution factor. There is no much difference in the second metabolite with the dilution factor here is, but the potential concern is still was why there is a high value with the sample is. So let's look at what we have done it. If you look at the chromatogram, it was showing very good asymmetric peak is. There is single chromatogram. There was no additional peak that can potentially lead that there is a merge of the peak and there is some additional chromatography is. So that also potentially shows that there is no issue with the chromatogram is. Then what we have done it, we have again go back and looked at a lot of the other literature, and we realized that the analyte which undergo into the biotransformations leads to the not only the two metabolite, but there is another metabolite called as O-desmethyl diltiazamine. And that is what in the initially I told that when the method comes to a bioanalytical scientist, they try to look at what the regulatory requirement is, and they try to focus on only those metabolites as well. They have ignored the third metabolite. So what we realized that probably there could be a contribution from the orthodesmethyl diltiazamine. Then it took a lot of the time to reoptimize the method. The chromatographic separation that took almost a couple of weeks time. Then there was a concern about the stability of the metabolite that has been fixed. The consistency of the result when we found that yes, my method is working now. We have separated two of the peak Cs. Then we have demonstrated with couple of the study sample reanalysis. I'll just show you. Now you can see it here. When we have applied this method, the method, the reoptimize again, revalidated them. That took almost a month of time to spend on the assay method. And then we realized there is a contribution from the O-desmethyl diltiazem that was exactly eluting along with the N-desmethylase. And that separation is not so easy task. That takes almost a month of time to separate it out. When the method is revalidated, that took almost 2 months time. And then everything was fine. But before the study sample analysis, it was also a strike it, you can see the data here. This is the initial data. You could see it here. Diltiazem, this is the N-desmethyl diltiazem. This is the initial subject data. When we have separated, we could see that there are two peaks. One eluted 1.93 minute RT, another is 2.5 minute RT. And the contribution of the 2.59 minute RT, which was potentially a O-desmethyl, contributed almost 50% of the concentration. When we combine together, you could see the percentage difference is very close from the initial value is. This is initial value. When we have combined these two peaks, the value is here. So that completely, they gave her thought that yes, there was a contribution from the another metabolite is. We have now, the reoptimize the method, and we are about to start the, the analysis. But immediately it was realized that no, let's look at the, the guidance, because in a current regulatory landscape, the agency keeps on changing, the guidelines is. If you look at in the OZD guidelines in 2019, the agency asked us to estimate both diltiazem and two of the metabolites. But in the June 2020, the agency asked to only estimate the diltiazem. You don't require to estimate desacetyl and desmethyl, and there is a valid reason, because this is there was the conversion between desacetyl and desmethyl into the body itself, and potentially during the in vitro analysis as well. And that's the reason agency has eliminated the estimation of both the metabolite. So the key learning from this study is whenever somebody is developing a method, or before going for a bioanalysis, let's look at if there is any guidance release, because we have spent a couple of months time to revalidate the method, reset up the method, the time consumption, runtime was very, very high. But at the end, what we potentially realize that we don't require to do anything. We just go and utilize our, the original method itself, because the original method runtime is just 3 minutes. And when we have re-optimized, the runtime is almost 10 minutes. So we again went back and utilized the original method. So only learning here is whenever somebody is going to do it, let's go and look at what is the current guidelines is. That would eliminate a lot of, the resources which is going to be used for your asset development, re-optimization, analysis, and the investigation. You don't require to do it, right? And then while developing a method, it's always good and the recommended that what are the metabolites reported. Let's look at if those metabolites are commercially available to see if there is any potential interference from those metabolites to the analyte, right? And this is one of, this is very, very common across any of the regulatory query. Agency always try to, emphasize that whenever the applicant submit the dossier, always the FDA recommend that, go and look at the agency current recommendation is, because nowadays the recommendation and those OZD guides keeps on changing it. So this was the second learning. Let me see the time if we have still.
Yeah, so we have another 5 minutes, Amarnath. Yeah.
That's fine. I mean, I'm on time. I can close it.
Thank you.
Okay, this is the another study which is very, very unique in nature. This is rivaroxaban study, which is again on COPD patient and almost, almost the 400 patient. The challenges in this study is this is the two moiety need to be estimated. One is the rivaroxaban, which is called as TD4208, and there is another metabolite. The challenge for this study is you need to go and detect the LLOQ 0.5 picogram. So as on date, we have the very high sensitive instrument available. Now the Sykes, which we have the 7,500 and all. But the challenges not only with the lower detection is, but the challenges with this study was the potential injector carryover is. It's not so easy to go and detect the 0.5 picogram level into the plasma is. So let's look at what are the things we have done it here. So the challenges with this drug product was the potential carryover with the drug moiety when we have, because the standard the standard 8 concentration is around 250 picogram. And when we have analyzed the subsequent sample, there is a potential carryover almost 47,500, which is very, very high from your LLOQ area counts is, which is almost, 9,900. And if you look at the carryover, it is almost 479 times carryover. The next subsequent sample blank, if you look at, again, the carryover reduces to 93%. And the third sample is still there is a carryover to 62%. And as we know, the carryover more than 20% is not acceptable at all. So what best the analytical scientists can do it, they will try a lot of trials. I'll show you. This is just a chromatogram. You could see the very intense peak for the higher concentration, the blank one. There is a carryover, another blank is a carryover, the third blank is still there isn't carryover, and this is very low concentration of 0.5 picogram is. What is the best the bioanalytical scientists can do it? They will try whatever the best they can do with the rinsing solution, the needle wash solution. This is what the best it was done it. There is a different composition is being used. A lot of the other trials, I mean, that is what the best, the one of the bioanalytical scientists can do into the bioanalytical lab is. The best it was done it, but in none of the cases, I have not presented all this data, but you can imagine that nothing is left to optimize, to minimize the carryover impact is. And none of the cases we have find that there is a further reduction in the carryover. The problem was remain same. In fact, not only the carryover, we have tried with the different mass spec, different UPLC, HPLC fronted, we have replaced with the injector port, degasser, the needle wash volume, even the HPLC vial with the glass PP tubes, runtime extended, isocratic, gradient, mobile phase combustion. This is what the best, everyone is trying to do it. Tubing material, injection volume, none of them workable. I mean, it was very difficult for a scientist to how to eliminate, and still because they need to get the 0.5 picogram is, and typically what people used to do it, if still they used to get the carryover, typically they just increase the LLOQ concentration from 0.5 to 1 or 2, because still they are getting LLOQ carryover. But the challenges for this product is you cannot further increase the LLOQ because these are the patient study, and there may be instances where you may get only the 2 picogram or 3 picogram Cmax value is. So what the best we have done it? We have again tried with a different gradient program, 2, 3, or even the more gradient program. We have also come up with the mitigation soil use where the right solvent we need to use it. We have also come up with the high purity solvent and the commercial water that can also potentially, the use to minimize the carryover is. You will surprise to know, I mean, I have not seen ever any of the bioanalytical lab that people are using, the multiple gradient program in the single run to minimize the carryover is. In this case, what we have done it, the total runtime was only just 5 minutes. And what we have done it, we have used the 3 gradient program. So the runtime we have extended to the 15 minutes. I could see the data here. This is a 5-minute runtime. This is the initial gradient program. The peak comes around 2.4. And we have another run the gradient program 2 and the gradient program 3 in the same analytical column. And you could see that there is a carryover in the gradient program 2 here, in the gradient program 3 here. And now this is a control bank. The response of this control bank is very, very low. And when we have run the very high concentration, you could see then there is a analyte which is retained into the column at the same RT2.4. And you could see that here, 7.4 minute and 12.4 minute. That indicate that there is still a moiety retained into the column. Even we have tried with a lot of the chemistry with the column. And that given a scope that if we use the 3 gradient program in the same analytical run, the data would be, I'll just show you here. Now you could see here, this is with the 3 gradient program. And again, we have used the methanol mitigation vial. I mean, we have tried with the different solvent and we realized that from the every samples, which is the typically analyzed, we need to use the methanol as a mitigation vial. So the carryover from the previous sample will get diluted, and keeping a 3 gradient program for a 15 minutes run time. Now it's a 15 minutes run time and the 5 minutes run time for a methanol that required, again a 20 minutes run time. And now you can see is from the ULOQ, you could see the methanol, the contribution is very, very low. This is 1%. And then the next sample, the contribution is 0. But again, this would be a very lengthy run time. The next steps, what we have done, but that gave an idea that it is difficult to pass through everything from the column. Then what we have come up with, just use the mitigation valve from the every analytical samples, you can see it now. The contribution from the standard 8, the methanol contribution is only 38%. And then the next sample here, we have just demonstrated if there is a control bank, the contribution would be just a 6.6%. I mean, this is a very, very unique approach where people are struggling to minimize the carryover. And if they are working with such a very, very low detection limit, this is the very unique approach people can adopt, and they can insert the mitigation vial for the every sample, and probably they would get, you could see the accuracy here, right? You could see the accuracy for every sample, standard 8, you could see the accuracy with the sample is, 100%, 97%, 99, 99. So accuracy is not going to compromise, your data quality is not going to compromise, but this is a very, very unique approach people can think about, and the work on this approach. That this is the new, the learning for us, that you can able to detect and quantify a very low concentration level by using this approach. You can further save an investment and time and the resources on developing an SM method for a month or so. There is no compromise in the quality as well, and this approach is still acceptable to the agencies. You can put an additional criteria for a batch acceptance, but nevertheless, your quality will never get compromised. This is another study. This is again the injectable complex product. Again, it's a multi-center study on a patient. Again, it's required a total serum iron, the transferrin bound iron. This is a very unique, the learning from this study we have. In this case, what we have seen is there is a multiple pharmacokinetic peaks for both the total iron and the transferrin bound iron. I'll just show you here. Let's say there is one subject. The concentration profile is like this, but all of a sudden you could see there is a dips in the concentration. Again, some of the point is very, very low concentration, lower detection limit. When we have reanalyzed this sample, you could see that the concentration is remain same, but absolutely there is no, this profile is not a PK data profile. Like another concentration, another data you could see here. Here again, you could see the PK profile. There is a sudden drop into the concentration. Let's see another couple of concentration. This is a transparent bound iron. The PK profile is very good. At the elimination point, you could see very, very high concentration here, right? There is another case. You could see there is a multiple dips and ups. 399 BLQ, then BLQ. It clearly indicates that there is some sample mishandling occurred either at the bioanalytical side or maybe at the clinical side. And again, this is a patient study. Let's look at, so what we thought that let's look at the performance of the assay method is. When we look at the performance of the assay for total iron, we could see that there was 124 runs and 123 analytical batches were passed. There was only one batch failure. The curve and QC performance is absolutely excellent. I mean, nobody can question mark about the bioanalytical data and the performance of the assays. The incurred supper reanalysis is also 100%. So absolutely, there is no concern about the bioanalytical assay method here for the total iron. Where it comes to the transfer in bound iron, again, the incurred supper reanalysis is 93%. Curves and QC performance is close to the 100%. The run failure is very, very low. That indicate the method is perfectly fine. There is no concern about the assay method is. We have taken a couple of samples which show the abnormal value. The data is very, very close. Initial value and the repeat value for the total iron, transfer in bound iron. That absolutely eliminate there was any concern from the assay perspective. We have investigated the clinical side. The clinical folks, they also did a lot of investigation. What they can do, they can only investigate based on the data, based on the documents available. For example, the IP administration form, the bleeding seed, they have interrogated the phlebotomies. They understand how the sample collected, process, shipment. I mean, whatever the best they can do at the clinical site, because this is the various site across India, they have investigated at every site. But the concern was only one or two sites, so they have tried to look at whatever the best they can do it. But the outcome investigation was nothing. I mean, based on the data, nobody has concluded what could happen, but it potentially raised a concern that there was some sample, the sample issues from one patient to other patient is. So what we have learned it from this study, what we have learned that, we should not 100% rely only at the clinical side, especially when there is the clinical trial or the patient study. Let's put at the third party QC monitor. And the third party QC monitor, the role would be take a picture of the vacuum before collecting the sample from the patient. Make sure that the vacuum ID is aligned with the sample which is going to be collected for the patient. So it would be a right vacuum with the right patient is. Again, you take the picture of the vacuum from the subject before the centrifugation, and then after the centrifugation, before a transfer of sample from the vacuum to the Rhea vial to making aliquot 1 or 2 to make sure that the right vacuum sample is transferred to the right Rhea vial is. So that again, those pictures can be go and verify by the people to make sure that the right sample has been transferred. And this is what we have put into the system. And from there onwards, we have not seen any kind of, the issues so far. And this is the another approach we have done it. You also go and look at the sample logs, volumes logs at the clinical side, how much the volume they have collected into the yeast tube. And the later you can go verify. For example, once you collect the blood, you take the picture of the sample. Before transfer, you make sure that all the samples, the ID is aligned. And then after transfer, you take the picture. This is what we have also tried to look at. What are the plasma you have transferred it? So the later, if there is any abnormalities you identify, you would able to go and cross verify if there is any sample swapping or there would be any mishandling of the sample at the clinical site. So I mean, this the additional control at the multiple clinical site would really eliminate such kind of issues, and which probably majority of lab used to know the gates, the find such kind of the issue is. So I thought that I'm trying to the cover in 40 minutes, 45 minutes of time. If anyone has any kind of the questions, probably they can ask me.
Yeah, so Amarnath, I would request you to stop the sharing so that we can display. Yes, great. And in interest of time, first of all, thank you, Amarnath. As always, you've been a great speaker and so much learning to, and then very precisely, all the case studies were very interesting. And I'm sure there are quite interesting questions also related to that and good learning for everyone. So in the interest of time, may I now request uh, the moderator Srinivas to introduce our next speaker, and after that we will actually take the panel discussion. So Amarnath is going to be with us, so we will have panel discussion then, and we'll be able to we'll be able to take up all the questions in the panel discussion. So over to you, Srinivas, and Shefali will take up the Q&A in the panel discussion. Thank you.
Yeah, before that, quickly, would you like to felicitate Amarnath?
Oh yeah, so since he's going to be there for the panel, yeah, he's going to be there for the panel discussion. So we will do for both the speakers at the end of the panel discussion, if it's okay.
Sure.
All right.
Yeah.
Thank you.
It's a great pleasure, I mean, to hear to Amarnath as well. And now moving on to the next speaker for the day. We have Mitesh Pillai. Can I have his profile?
Yes, please.
So Mitesh, again, it's again a pleasure to have you back on APA speakers list. And, Mitesh is actually currently Deputy General Manager and Head of QA GLP Department at Zadis Research Center in Ahmedabad. And he actually has been quite active in the quality field, and he is exemplar global certified Six Sigma Black Belt quality professional, and he's also pursuing his doctoral actually in business administration from SPJN School of Global Management. His area of research is digital transformation. I'm sure he has a lot to mention since he's talking about digital transformation. Today is also going to talk about reduction in failure rates in the regulated bioanalysis, the human error perspective. So I can relate his area of research interest and what he's going to discuss with us today. He also completed his executive program in management in leadership and change management from XLRI. Actually, he received his MSc in industrial chemistry from Sardar Patel University, and for last about 17 years, he has been working in the field of research quality assurance. So he has faced multiple regulatory inspections and has a clear understanding what regulators can expect from a quality system. So just to mention, US FDA, MHRA, ANVISA, as well as OECD GLP were some of the inspections that he has faced successfully. And he has overall knowledge of GXP quality systems, and currently he is also interested in developing technology-driven quality tools which shall address the current needs and issues of pharmaceutical industry. So with this, Mitesh, I welcome you to deliver your talk.
Thank you. I will be sharing my screen.
Yep. Yeah, it's visible.
That means the screen is visible to everybody?
Yes.
Yes, it is visible. Please proceed.
So I would like to first thank Dr. Srinivas Sadle and Dr. Deepak and everybody in the APA governing body for inviting me for the presentation. Today, as Dr. Amarnath has clearly explained the root causes and the reasons for evaluating the failure in 4 case studies which he has explained. So I would be taking it forward from there, how we can reduce the failure rates. I would be talking here about the human error perspective. So the areas which I would be quickly covering is just an introduction, the root causes in bioanalysis. It would be a common industry practices. I would be also discussing what is regulatory expectations, then a small case study from aviation industry, a loophole in the current practices. Then I will talk about the quality system approaches, then how we can manage human error and how we can reduce it. And I have proposed finally a 6-step methodology. So to introduce, Humans are not perfect. They do mistakes. And that's the core part. Most of this error lead to major failures. So in average businesses, it is less around 10 to 30%. The human error rates are there, which lead to the failures. And in a best quality practices, if we implement, still there is a avenue where we have 5 to 10% of the same. So we cannot completely remove it, but it stays with us, but how we can mitigate it. So today I will be discussing something about the identification, rectification, mitigation, and control of practices. So with this, the core failures of the core reasons of batch failure, that is, one is a sample preparation error, sample processing error, bulk spiking error, incorrect method selection during analysis, wrong solution preparation, and the most critical is the in-cut failure, which lead to maximum causes of going back for a method development stage, and the repeat analysis. So what is the common industry practices? What we generally do is that we do a documentation, we do a risk assessment, and we have an investigation plan in place, and we do and mitigate the same. So we have accurate procedures, and we try to enforce it and try to check that our procedures should be accurate. And finally, we do a training of the people, just as a CAPA and the preventive measure, and we try to mitigate the thing. But what's the regulatory expectation? The regulators want all the failed and the past reports of all the things reported, because they can evaluate it. They are asking for 100% result summaries of past and failed report. All run statuses, pass, fail. They want to evaluate what is the cause behind and how we have investigated it. And also the table of calibrated concentration, including both pass and fail. And ISR fail investigations also. So this all lead to a common perspective that failures are critically taken by the regulators, and they do a proper evaluation of the same. So with this, I would like to come up with a trend of 483s. This is not related to, apart from bioanalysis, it is for the entire industry as a whole. You can see till 2029, the rate is increasing, but due to COVID, the inspections were less. That's why the 483 rates are less, but there is an upward trend in the 483s. I have divided that based on the country-wise perspective. So India and China contributes the maximum. Then comes, I have done a Pareto analysis to see that what is the cause of this warning letters and 483s. Basically, it is inadequate investigation. And the second is the role of quality control and the quality systems. So these are the two things which are contributing maximum to build this. And in this, I have also tried to find out what are the contribution of human error. So we can see here from this Pareto graph that the maximum contribution is from the non-compliances of part 11. Second is the falsification of data and retesting without justification. So falsification, retesting, recording, this all falls under basically human errors. And these are leading to basically contribution to the warning letter. With this, I would like to take your take you through a case study which I have evaluated in an airline industry. Why I have took airline industry? First, to explain, it is a highly quality conscious from a functionality point of view. They have implemented highest quality standards, because if a error happens, if a human error happens, it directly leads to loss of life. So I critically took this evaluation from a website where it is very clearly shown that in 1903, the machine causes fatality was 80% and human causes fatality was 20%. Today, the human causes due to any error is 80% and machine fatality causes are 20%. As we have technologically advanced, the machine errors have reduced, but the human causes have human errors have been increasing. But if we can see, the fatality index is almost reducing. Number of accidents are reducing, number of fatality indexes are reducing. So what differently they have done? What they have done is that, if I can say, tell you the sensexes, in a plane oddly a person die as per the US census data in one passenger in 2 lakh, and the dying rate is 1 in 2 lakh. But in other cases, just like cycling, it's 1 in 4,000. In drowning, it's 1 in 1,000, and in car crash, it's 1 in 102. So what they have done, even though there is a ratio of human error is 80%, accidents have gone down. So what they have did is that they have used new technologies which can prevent or mitigate the human error. Second, they have implemented some good quality checks where that if an error happens, that it does not directly lead to failures or a fatality. So with this, I would like to draw everybody's attention on the current practices which we are having for the investigation part here. So here what happens is that we don't have a standard approach to dig down on the error in human. We have an inappropriate root cause and inappropriate kappa processes. And there is a misconception generally that due to improper training, human error happens. So in any investigation, which was discussed in earlier presentations also, we need to have a proper root cause analysis. Just like the last last case study, which Dr. Amarnath has told, that they found an issue at the clinical site. So I am just sharing a root cause processes that we need to have the failure and evidence of proof. Evidence of proof in sense, if we can correlate that he told to have photographs of the samples. So we need to have a proper evidence of the proof. Then we need to have a proper investigation and understanding of the root cause. Based on the identification and understanding, we can do an analysis and a corrective action plan. And based on that, we can have a proper implementation plan, that is a project management plan. So what are the techniques of doing root cause analysis? One is a brainstorming section. Second is a fishbone, that is generally used as fishbone, 5 and I technique, kappa processes, and Pareto diagram, and a failure mode and effect analysis. Basically, most effective is the fishbone, which generally is used internationally, and we can use other modes of also of root cause analysis, but we should have a proper methodology of identification of the root cause. With this, I would like to take you to a journey of quality assurance, where it was just first, it was a, there are 3 phases of thing. One is reactive. Reactive is basically based on the observation we need to be used to improve. Our quality system used to correct it. The second was proactive, where based on guidance and the past experience, we used to correct ourselves. And then comes the future, which is going to come in near terms. Already in manufacturing, in pharma manufacturing, the things have started implementation. But I think in few years it would be into bioanalysis also. That is a collaboration between human and machine, that is predictive. That means that we are predicting the possible error and mitigating it before it actually creates a problem. So already the airline industry is into a predictive stage. I think in few years the pharma would also be using there, going and reaching there. With this, what is basically predictive? Predictive means generally human interventions are linked to subjective biases. How to avoid that? So AI can play a critical role in doing that. We can give AI a task of doing repeated things. In Industry 4.0, generally the trend is that the entire blockchain AI can be used to manage the entire processes. But in the current scenario, we all have a lab which has a historical data of almost 10 years, 20 years. We can have an AI solution which can deep dive into the system and try to find out the possible issues and cause. And then using AI, it can tell us the root cause. And once it learns it from the past, if you have a scenario, it can help you in finding out the future issues also. And second is the automation part, which I have discussed. We can have lesser error by digitizing the entire processes. Second is where we are currently, most of the biomedical divers started doing it. Some have reached a certain level. We can reduce the manual deviations. We can reduce the deviations by removing all the manual processes and capturing the systems online. And we can have a faster review process. With this, I will finally reach to a place where how we can reduce the human error, how it is a management. This is a general flow. If a human error is identified during an investigation, there may be two causes. It may be a system problem. It may be a human performance problem. If it is a system problem, then we can have some administrative management systems or a software or a process in place where we can remove it. But if it is a human performance problem, there may be two factors which may be impacting. One is the working environment. That is external factor. If it is an external factor, we need to correct it. Or if it is an individual issue, then we need to work on the cognitive loads. That means that if it is a environmental or a system issue, we need to work on the factors to improve that. But if it is an individual issue, sometimes we identify it as an individual issue, deep dive into that, then we need to work on the human characteristics of that person. With this, I will be proposing a 6-step reduction methodology if it is a human error issue. First, we need to diagnose. What is really mean by diagnosis? Diagnosis means that we need to discuss with the team, with the people, with the persons who were involved in the activity, try to identify what went wrong, how it went wrong. And it's due to a human nature, or it is due to a process issue, or it may be due to a circumstantial issue. Then based on that, we need to have a training and coaching program. Generally, currently what happens is that once we identify an investigational issue, we do directly jump into a training and we do a SOP training or a protocol training and we think that it's closed. First, before having a proper training and coaching, we need to first have a diagnosis process in place where we need to try to fix on that what's the problem. Based on that, the training and coaching needs to be done. Training and coaching is not only related to SOPs and practices, it should be on behavioral part also. That means that how we can improve the behavior or how we can try to improve the mindset of the people. Based on that, we need to have a human reliability assessment. That's a very critical parameter which is coming up nowadays because most of the data integrity issue across the globe are due to the reliability issue. Somewhere is due to reliability issue. So we should have a setup in our lab and in our processes that how we can check the reliability assessment, how we can do that. Based on that, we need to have some system change implementation where if we really feels that the system needs to be changed, we need to implement that in our system, which will lead to a cultural change. And once the cultural change happens, automatically your error rates goes down. And once the error goes down, you have a lesser chance of rework, you have a better quality, and the most important fact is that you don't have to create so much of investigations, deviations, or and record it into the reports, which finally leads to a issue to the regulators. And then we need to, after the cultural change happens, we need to have a monitoring and trending of the same. So in these 6-step processes, which it helps in reducing the entire process, entire human error. It cannot be diminished, but it will help in doing that. And this is a concept which is definitely taken from a human psychology point of view. But if implemented in a quality system, we can definitely achieve the reduction. With this, I would like to show. I would like to conclude that there is a paper which was published in 2018 which told that if we have proper training and an automation, the reduction in failure rates has gone down by below 3%. That means that a lab which has tried to do this training and automation, their failure rate has gone down by below 3%. Less than 3% is a good number. This will help us in having better resource planning, better productivity, and a quality data. With this, I would like to conclude with this a small picture, which I started off with the slide. this is just a human and machine collaboration. It's not like that machine can replace human, but they work hand in hand to help us. Just like LCMS instruments help us in analyzing and giving us the result, but the analysis and the sample preparation is done still by the analyst. So it's not a replacement of the people. It's a environment where work machine and human works to create a better result and a better quality. With this, I am concluding. Thank you.
Thank you, Mitesh. Very crisp and focused talk and very interesting findings you have shared. Let me invite the moderators and the earlier speaker, Amarnath, also, and for the panel discussion. And would request Mitesh if you can stop sharing the screen so we can display
I will do that.
Yeah, thank you. So over to you, Shefali, Patanjali, and Srinivas to take up the questions with both the speakers. And while you take up the question, I would request Mitesh, if you can just go to the virtual background, and if you can select none, it would slightly help, because it is sometimes it's coming off your face, so we are not able to see you properly. Yeah. So if you can just remove the virtual background, that's also fine, and even this will work. And if this, yeah, this is perfect. Yeah, great. Thank you. So over to you, Srinivas, Patanjali, and Shefali. And Shefali, if you can just switch on your video so that you can also be on the screen. Thank you.
Yeah, Mitesh, that was a great insight. Now we see that technology is entering in all fields, and to the best use of it, we should really deploy technology to minimize human errors. With this note, I'll just open. There are a few questions already that have popped up, so I'll take one by one. Maybe I'll start first, and then Patanjali and Shefali come up. So there's a question from Karthik. He's asking about ANDA submission. Do we need to develop and validate formulation-specific bioanalytical method? We believe that the rate and extent of absorption expected to be similar in comparison with RLD. We always have wide CC range which could cover any dosage forms and avoid duplication to reduce the cost and resources. So what are your thoughts? Do we need to go for a formulation-specific EMA? Amarnath, maybe you would like to take that.
Absolutely. So let me clarify them. He can absolutely go and validate the SM method with a very, very wider range. Nothing is going to stop them. Agency would never ask any question why you have validated your curve range with a wider, the calibration with the LLOQ and ULOQ. But the important point here is, let's say if you have the various dosage forms, the various dose strength. I think his question is, let's say if you have the formulation with the 10 mg dose to the 100 mg dose, right? You cannot validate the method with the 100 mg dose, with 50 mg dose, and the 10 mg dose. You can absolutely validate your SA method with a wider calibration curve range. But when it comes to the study sample analysis, you need to choose a curve range which is a very, very appropriate and accurate for your study sample analysis. Let's say you have validated your range considering a 10 mg dose to a 100 mg dose, but tomorrow you have a 25 mg of dose formulation for a study. You can reduce your ULOQ concentration. Accordingly, you can spread your quality control sample as well, and then you can do the study. So agency will never have a problem as long as you have a minimum 2 or 3 QCs within your expected cover ranges. So in such cases, I mean, we do routinely. I mean, being a pharmaceutical company, we have a lot of formulation with the various dosage forms. So what we used to do, we validate a SM method with a broader range. And when it comes to the study sample analysis, we appropriately select the curve range, and then accordingly we spread out the QC concentration. It's not necessarily that you have the multiple validation for a multiple dosage strengths.
Sure. Patanjali, would you like to take the next one?
I have a follow-up to Amarnath. In that case, when you truncate the curve, right, you do a 1ANP run just before you touch the study sample analysis?
Absolutely. There would be a 2 precision accuracy run. And that is, I think, in one of the regulatory, the review from the EMEA, they have asked that only 1 precision is not sufficient. You need to have inter and intra precision. So whenever you are going to put additional QCs or truncated your molecule concentration, you need to have at least a 2 precision accuracy. I mean, this is one recommendation from the EMEA, but FDA has never asked that you need to have a 2, but it is always good to have a certain accuracy and precision to make sure that your truncated curve is still going a very linear curve range, and your additional QCs are your QCs when you are going to change it, giving a good, precise and accurate result. So wherever we work with the CRO, we always tell them when you are going to truncate your curve range or when you are going to put an additional QCs for at least for mile and a weight trace study, you need to have a 2 precision accuracy run.
Okay, there is another question from Avinash Kumar. I think it's to you. I think in discovery stage of development, where concentration is not known of any analyte, how to keep the dilution DQC to answer any regulatory bodies?
See, the challenges for where the concentration is not known, probably in such cases, people used to take a very wider ranges, right? And during the wider range, and if still their concentration from the animal data is very high from beyond the curve ranges, still they can go and demonstrate their dilution integrity, the fold which they are going to utilize for their animal sample is. So as long as your control sample dilution integrity is within your curve range and showing your the 100% accuracy are within the 85 to 115, the agency would never point your any of the data, the integrity, as long as your dilution integrity is proven with the dilution factor being taken for your study sample is. And yes, of course, there would always be a challenge from the animal study where the concentration is not known. You can go with 100 folds, 200 folds, provided you also need to demonstrate with your control sample those dilution factors.
Hope that answered your question, Avinash. So if you have any follow-up, please put that in the chat if you need to be. So I think Mitesh has sent it something. Thanks for sharing on digital transformation, specifically AI for reducing human errors. This is for Mitesh, I think from Srikanth Dittakavi. So on a general note, if team is clear that we need to perform in all aspects of study team can minimize these human errors, that culture is the one which we need to develop in lab, I guess. Any comments?
So I think that generally in any steps we have all the SOPs and protocols in place. Still, we can see from the case studies which we have discussed today, the error happens. With specifically, if I will like to tell is that from the clinical site which Dr. Amarnath has told, there also the protocol training would have definitely be happened. So I think that it's not about the SOPs and procedures in place. We need to have a proper root cause and then identify where that error happens. If it is a human error, then we need to work with people to understand what is the problem. Whether is just for an example, if we have a process in our place that there is an SOP, and you just ask a person to demonstrate what is the SOP versus practice, you will definitely identify just a small experiment which I would like to suggest to you to do in your lab, that there is an SOP which is existing for last 2 years. And if you ask an analyst to perform that SOP versus practice in presence of everybody, you will find n number of issues that few of the steps actually is not required or it is overdone. So on ground, sometimes what happens is that steps are bypassed by human. No doubt it is written in SOP. If QA is there or any other person is there, it may be identified, it may not be identified. So what happens is that it gives to a standard result. Whenever the issue is there, when the results goes out of deviation, we go for an investigation. But actually what is there should be a culture in a lab where people should come collaborate, discuss their problems openly, try to identify if it is a human error, they should not be punished, they should be rewarded. and they should come up with the problems upfront to the management so that we can fix it. So it's a culture. You are building a culture. So it should start from a small experiment, if it is SOP versus practice, which I would like to suggest. So you can definitely, if you do 2 to 3 SOPs in your lab, you will definitely find out finally where the parameter of practical versus theoretical stays. So definitely, I believe that if you improve the culture of a lab, you will get a drastically good results. And that for culture, human are the most important part.
I'm 100%, they agree with Mitesh's thought. SOP is one part, and the practices is another aspect to look at. Every lab, every clinical site has a lot of the controls and balances in the place. It is well defined into the SOP. When it comes to the practices, in a couple of instances, we have seen that, the SOP versus practices is one of the culprit. Like I just shown in one of, the case study where, as we know, that the protocol training has been given, all the individuals has been trained on the procedure and practices. And couple of times we have seen that the sample sweeping at the clinical site. And what we have put in a procedure, though it's little an expensive process, is we have put an third party, the QC monitor. Their job is to make sure that, the right vacutainer has been taken with the right persons, the right sample has been transferred to the right tube, and their job is to take a pictures along with those samples together. And with the past 1 year or so, we had an experience that we have not come across with any kind of the sample sweeping, potentially, which is quite common in the clinical side. Similarly, when it comes to the bioanalytical aspect, yes, there is a procedure in place, there is a checks and balances, but still I would, suggest other lab, they should have the very, very robust procedure in the place instead of the SOP. SOP is yes, it's a Bible of any organization, but the practices is very, very important. And people, when it comes to the practices, people become a mechanical kind of the work. They feel that this is a routine work, and the moment they made one mistakes, the entire studies, they went into, the wrong direction. Then people spend a lot of time the investigation, incident, and even of because of the one incident occurred it. So the culture is very, very important. People to be very, very encouraged, and told them that this should be very, very transparent. If there is any error, because we know if the people are going to work in the lab, mistake is intent to happen. You cannot control it, you cannot stop it, but they should be fair enough that if the mistake is happened, they should brought to the management, tell them that this is a mistake happened. Nobody is going to punish them, but that would give a learning for others that this mistake has been brought to the knowledge to the leadership, and that the better control can be put onto the place.
Yeah, just to follow up, and nowadays there is no intentional mistakes happening, right?
Absolutely.
Earlier on, the intentional mistakes led to integration SOPs, chromatography SOPs, and all those SOPs came into play. People were playing around with that peak or whatever it is. Nowadays, it's all accidental mistakes. So as long as you can control those accidental mistakes, I think things will be. Further question I have to you and Mitesh and to Amarnath, if I can take a little time, Srinivas. There is an Alcoa principles adapted by FDA. You guys adapt those things in your labs? Respective Alcoa stands for attributable, legible, contemporaneous, original, and accurate. And there is also Alcoa Plus, which has available, enduring, consistent, and complete.
Yes, it is being followed across, I believe, across all the lab here in India.
Okay.
Yeah. So to add to the Dr. Amarnath, it's a basic requirement that it has to be followed because any data which is generated needs to have this characteristics DNA of Alcoa. Then only it can be accepted by regulator. So we, as a bioanalytical lab, since last 20 years used to follow this, but finally the FDA has coined a word. Regulators have coined a word to relate that. But generally, GLP tells that raw data should reflect the report. That means that raw data should be accurately captured. And that was critically followed since I started my career as a bioanalyst. So Generally, its practice followed, especially in India, it's followed. Across the globe, it's followed.
Good to know that. Thank you. Any Shefali, any questions you have? You're on mute, I think. Deepak?
No,
Patanjali,
I'm good.
I have seen one question from Jayant. I think he was talking about the loading of the transparent bound iron. So his question was, instead of 1 cc, did you try with the 3 cc? So I just wanted to make him clear that yes, the 1 gram, the bed capacities comes with only a 3 cc, the SP cartridges. Even we have a try to look at the bed capacity with the 2 gram and 3 gram. Unfortunately, this 2 gram and 3 gram bed capacity is not available commercially. The another the hurdle with the 2 gram or 3 gram cartridges is the size of the cartridges would be a 6 ml. Again, 6 ml would not be a true sense to use for typical with a large amount of the sample preparation. If you are in the R&D with a limited number of sample, probably you can do with the 6 cc of cartridges, but 6 cc for a large number of quantity of the sample handling would be very, very difficult. That sample is almost more than even 6,000 sample. So yes, we did with the 3 cc only, but 6 ml cartridges will potentially not feasible for any of the bioanalytical labs to handle with the large quantity of the sample.
Okay.
Sure, I think there have been many questions, and I'm sure they will also have additional questions. With the interest of time, I think, Deepak, we would like to take this session for conclusion.
Yes. So before we,
Sorry.
Before we officially conclude, Srinivas would like to felicitate both the speakers, and then I would request each one of you moderator for your concluding remarks. So I think starting with Shefali can felicitate Amarnath, and yourself or Patanjali can felicitate Mitesh. So let me just.
Yeah, thank you, Amarnath, for the, for always making it so enjoyable, for making your talk so enjoyable. You make it very real time, very participatory, and it's a lot of fun. Thank you for the lovely talk.
Thanks, Apali. See, I always try to spread the knowledge which we used to gain it, not only from our lab, but the lab which we work globally, try to get some of the real information which can really help us to other lab here in India. They can start doing such a practices, minimize those kind of the errors, and that would really eliminate a lot of queries from the agencies.
I'm not sure whether a couple of years ago I was talking to while he's presenting your,
Yeah, thank you, Amarnath, for a wonderful talk. And always, I think the theme, the talk was completely aligned to the theme of regulatory challenges and learning. So thank you for a wonderful talk once again. It's always been a pleasure to have you as one of the esteemed speakers for the conference. So thank you, Amarnath. Would request now Srinivas to felicitate Mitesh.
So Mitesh, it's an e-certificate though, but we really appreciate your contribution. You have always touched upon the quality aspects of RegBio, and today's was one such which is keeping in time. So we see that there's going to be a lot of owners, even smallest human error can really. And I think aviation and pharma industry is not very far. We also deal with fatalities if we do certain blunders. So thanks for touching that, and thank you once again for being part of the APS.
So thank you very much. And the future is basically the collaboration of human and technology. So I think we would certainly see in few years that how technology enters the bioanalysis arena also.
Yeah, I have a final quote, if you may allow.
Yeah.
To err is human, but there is AI to help. You know.
So to add this to one more point is that as we have lots of data in bioanalytical lab, we should start working on creating some AI because data helps it train. So data is the source for AI. So
Yeah.
if as a lab, if we can start working on that, you can definitely get a greater insight from the tons of data which we have in our labs.
So
Yes.
we
Let me, I think somebody's, okay. So let me quickly then request officially all our moderators. And before that, I would like to thank you to all our three speakers, starting from Clarinda, Amarnath, and Mitesh. Great talk, very interactive session. And as always, I think regulated bioanalysis is the longest session in our meetings. So we are close to around 4 hours now, I guess. So it's really a great interactive session. And let me now very quickly request each of the moderators to officially present their concluding remarks, starting with Patanjali.
Yeah, thank you. I think Clarinda is not there. I thank her very much. It's her presentation was a gamut of coverage. I think covered all aspects of bioanalysis. I think we might get her as a repeat speaker for the cut point establishments and false positive rates and things like that. And I also thank our good friend Amarnath Jaiswal. Maybe I don't know whether I can say is our actor Danny Denzopa. Reminds me of that actor all the time when I look at him, you know. And then also Mitesh for his enlightening talk about human error and recognizing that how human error contributes irrespective of how many controls you have, you know. It's almost like 2:15 or 2:16 a.m. here in New York. So I'll say thank you all and good night and see you next time. Okay. Thanks a lot.
Thank you. Special thanks to you, Patanjali. It is like 2:15 is real late in the night. And I must appreciate that credit goes to your wife. She has allowed you to be so awake so long. So thank you, Patanjali.
No problem. Thank you, guys. Have a good day. Yeah.
Bye-bye.
Thank you. May I now request Srinivas for his concluding remarks?
Yeah, I think today's was a great mix. We looked at small molecules, large molecules, and quality. And when it comes to regulatory challenges, yeah, I don't think there is any demarcation. And Clarinda really touched upon the current understanding of establishing biosimilarity, what challenges probably most of the innovators or sponsors are facing in that particular space. Amarnath, as usual, has touched the bioanalyst heart, and he always shares his experiences, and because he is really placed in a way as a sponsor, he also deals with a lot of biomedical labs, and the rich experience comes in front. I think APA is such a platform that where this kind of enriches all the people who are participating in this forum. And Mitesh, of course, earlier, gone are the days when we used to call quality assurance guys as inspectors. We used to just danda the team. That's not the case now. And everyone, when you say you are also extension of a team and trying to minimize errors from all aspects. So all of us are contributing in the overall process of bringing quality medicines to humans. So thank you so much, all the three speakers, for addressing all relevant different aspects of this theme today. That's it from my side.
Thank you. And finally, may I now request Shefali for her concluding remarks. Shefali? Shefali, I think she was just there. Shefali, if you can hear us, just requesting for your final concluding remarks.
I think she has some network issues. So
yeah, it looks like there's some network issues. So anyway, so thank you all the speakers, all the moderators, Srinivas, Shefali, and Patanjali for the wonderful job they have done as a moderator and taken up all the questions. Thank you to Amarnath, Clarinda, and Mitesh for a wonderful session. So with that, we come to officially we come to the end of this webinar. And let me now quickly launch the poll. So webinar poll. So would request all the participants to please participate in this. For some reason, I'm not able to launch the poll for webinar 10, but what I have done is webinar 7 poll is right there in front of me. I could access that. So I'm just launching that. The only difference is there's a question number 4. There are only 5 questions. So for question number 4 says the webinar talk you like the most, where because it belongs to webinar 7, there are only 2 talks, but options are 3: talk 1, talk 2, and both talk 1 and talk 2. So would request participants to consider this as talk 1, talk 2, and talk 3. So you can choose any of the answers. So in that case, we would be able to use the same webinar poll. So except for that, rest all the questions you should be able to answer it. I mean, submit your responses. So I'm just launching it, and there it goes. So it is launched, and you can submit your responses. Once again, there were 3 speakers, but on the 4th question, there is only 2 speaker options. But the 3rd option is both 1 and 2. Please consider it as talk 3. Yeah. So we'll just keep this for a minute and would request everyone to please submit your responses on the poll. I think even the panelists can participate in this. I could see an option where panelists can also participate. So hopefully you guys can also submit your responses. Even 2 of our speakers, Amarnath and Mitesh, you can also submit your response if you wish to.